Good morning, class. Good morning,
Good morning.
my wonderful people. In fact, a little bit sorry for late start a little bit because yesterday funny thing happened in Cape Coast here. Our power went out. It came on in some places, but some of us were not lucky. But we thank God that I'm able to see my wonderful people today. A little bit of shout out to my ladies before I can start the class. You know, this class is a little bit of a tradition. Oh, okay, Angie is here. Okay, so Kikeli, I see you. Alexi, I see you. Angie, Bright, Charity, Raymond, Dorcas, Eben, Edward, Emil, James, Jaden, Josh, okay, Lord, Mercy, oh, King Muhammad, oh, one, Ayub, Nash, I see you, Nico, I see you, Nuruddin, I see you, Pat, I see you, Paul, Piero, I see you, Phillips, Becky, I see you, Ritual, Rita, uh, Samson, Sammy, uh, Piero, Tia, Piero, I see you. Willie, and then Wisdom. Okay, I see you all. Okay, thank you for making it to the class this morning. I think a couple of people still are on the process of joining us. Some people are still waking up to the hangover over the holiday. Okay, but I'm happy you guys are here. Okay, good to see you here. All right. Oh, Tampori is back on fire here. I see him. He's back on. That's good. Good to see you. Good to see you. All right. So today we are actually going to wrap up the cell biology, and then so things that we intend to do today is that we are wrapping up the cell biology, and then after that we'll talk about the cell cycles, and then maybe we'll start with DNA. We'll go into the molecular biology a little bit. Okay, we'll go into the molecular biology a little bit. All right. So today, in fact, today what we're going to talk about is very, very interesting, and so we'll kickstart. Oh, Nimatu is here. Nimatu, good to see you. Good to see you. Welcome. All right. So quick recap of the last time. So the last time we did a number of organelles and stuff like that. So today we will wrap up that. Okay, we did some true or false. Those of you who couldn't join us, with a little bit of true or false here. Okay, I think this is the old true or false. Okay, so we did a number of them. Okay, and today we're going to continue from where we ended the last time. Okay, most of the points here we've done with that. Okay, good. So today, what we are done with the endomembrane system. So we will talk a little bit of the non-membranous organelles. Okay. All right. So let's start with let me start from here.
Good
Okay.
morning, Doc.
Good morning. Please, how are you?
Fine, thank you, Doc.
Good to see
Please.
you. Okay. I hope all is well.
Yeah.
Oh,
Yeah.
yes, please.
Listen,
I'm listening.
we are here to get the slides for the last lecture.
Yes, yes. So I needed to add the one we're gonna do here today.
All right.
And then you see all the slides. Do you know all the slides? I've given all the bulk one to you. Just that one.
Yes, we have it.
Yes.
We have it, but because you always write and
yes, please.
and add some things,
Great.
it makes it more understandable.
Okay, well, that's great.
So we like those ones too.
Okay, yeah. So please, I decided to add a little bit of some trial questions and stuff like that. Okay, so I want to do that one. Then I will send all of them to you. Okay.
All right. Thank you.
You're most welcome. All right. So Okay, so today we'll deal with the non-membranous organelles. Okay, so what we're going to talk about organelles that are not bounded by membranes. Okay, but there is one membranous organelle that I need to talk about. Maybe you may not see it in your slide, but it's very, very important. Okay, so let me talk about it briefly. Okay, so that's a peroxisome. Okay, good. All right, this is a special. It's a membranous organelle. Okay, it's a very special membranous organelle. Okay, and and the reason it is very special is that it is not strictly that type of word. In fact, what it does is not really following those ones that we know. Okay, usually it is less mentioned, but in fact it carries a lot of metabolic activities within the word, within the cell. So I need to talk briefly about it. Okay, so I don't know whether you've heard of it before, but in case you've not heard of this one before, this particular organelle, okay, uh, it is single membrane bound, just like the some of the others. So it is what? Yeah, it is spherical, almost spherical, and was membrane, single membrane bound. Okay, and this one it contains what? It contains um some sort of what enzyme, but there's one particular enzyme which is so important. Okay, it contains an enzyme, we call it catalase. Okay, now do you know why these catalase, if okay, before even I okay, enzyme called what? Let me see. An enzyme, an enzyme called catalase. Okay, and what is this catalase going to do? This catalase, what it does is that there is this particular substance. Okay, we call it what? So let me say it again. We call it hydrogen peroxide. Hydrogen peroxide. So the formula is what? H2O2. In this hydrogen peroxide, it's a very dangerous chemical in the cell. It is you see, it is having something we call reactive hydrogen. And hydrogen peroxide, if it leaves our cells, okay, if it leaves our cell, it's a disaster. Why? Because it will destroy our DNA, to destroy the cell membrane, to destroy some proteins and stuff like that. But the question is, how come this hydrogen peroxide at all appear in those in the peroxisome? Okay, how come? How come? That's even the first question to ask. How how did it get there? How did it get there at all? Okay. Well, now the thing is that we have something. Okay, so before I okay, so this peroxide, okay, because it is then I'll talk about how it get there. Because it is dangerous, it needs to be broken down. And so how am I going to break the peroxide down? I need an enzyme called catalase. Okay, this enzyme called catalase. And what this enzyme do? This enzyme is going to break this. So it's what? It breaks. Okay, it's what? So it breaks down. Breaks. Okay, it breaks down. It breaks down the peroxide. Okay, the peroxide. All right, the peroxide into water and oxygen. Oxygen. So we by this formula, let's say this one H, so 2H2O O2. Okay, all right, like that. Okay, giving us what? What is it going to give us? So H2O, like that. Okay, so it will break down to give us what? To give us water, 2H2O plus what? Plus oxygen. So O2. I think it's balanced. Okay, yes. So this is what we're gonna get. Okay, so and this is normal oxygen. Okay, so this is the work of what? So this is what the enzyme, this is what this enzyme catalyst does. Okay, so let me put it on top here. Like that. So this enzyme catalyst, that's what it does. Okay, it's what it's actually breaks down the hydrogen peroxide that I said is very dangerous. When it breaks it down to water and oxygen. Okay, so this particular organelle is very, very important. So it contains this enzyme we call catalase. And what this enzyme is that the enzyme that's what this enzyme breaks down hydrogen peroxide into water and oxygen. Okay, now the question that I asked earlier, how did it get there? Okay, now this hydrogen peroxide is regarded as something we call reactive oxygen. Okay, this when it escapes is dangerous. Okay, but how did it get there? Okay, what happened is that a very long chain. Let me take this one. Okay, so what happened is that this very long chain where fatty acids. Okay, so let me get you a picture so that makes life easy. Okay, so let's see. Uh, okay, great. Let me get a picture for you so that you get a rough idea of what I'm talking about. Okay. Anything. Okay, so let me take. Okay, getting very good image. Okay, okay, let's see. Okay, maybe this one. Good. I've gotten a very beautiful diagram here. All right. Okay, great. Okay. All right, here. Okay. Yep. Okay, so let me.
So
Yes, please.
I think that you're balancing. You left out something. The oxygen gas should be balanced by
Is it
putting.
not balance? Let's see.
No, there should be two in front of behind the oxygen
Okay, let's see.
in the
Wait,
product.
let's see. So this will give us 4.
Yes.
This there is 2 here.
Balance.
It's
That
balanced.
is balance.
It's balanced.
It's
It's
balance.
balance.
So 2
It's
times
balance. There's
2,
no
4.
problem with that.
Hey,
2
Water
here.
has
It's
it's
balanced.
already balanced. Oh,
No.
It's
Oh,
balanced.
okay.
It's balanced. Oh,
Oh, sorry. Yeah, it's balanced. It's balanced. I just saw the two over there. Sorry.
Yeah, we have a lot of chemists here. Hey, you, my ladies, it's not balanced. I
It's
didn't hear the
balanced.
balance. No, the balance. I didn't hear voice of the lady. Hey, what is happening? 'Cause my ladies are doing chemistry as well. It's balanced.
balance. Oh. It's
Yes,
balance.
yes. One lady has spoken. That is it. Please, I won't even mind anyone. Don't mind them. Don't mind them. Hey, they should get revise the chemistry notes. It's balanced. One lady says it's balanced. So it's balanced. Okay. All right. Okay. So look at this diagram here. Okay. Look at this beautiful diagram here. Now, so it is single membrane bound, as you can see from here. And there is something here. Look at. So what happens is that you see, if you look here, we have something we call long chain fatty acid. Okay, long chain fatty acid, and then we have something we call branch chain fatty acid. All right, so what will happen is that we have something we call alpha oxidation and beta oxidation. Okay, so what will happen is that so when we have very long chain fatty acid, so okay, we have very long chain fatty. So let's say long chain fatty acid is like this. So let me get you let's see, can I draw? Let me see, I can draw here. All right, great. So here we have something like this. Okay, this very long chain fatty acid. And this long chain fatty acid, let's say I've drawn here. Basically, I can't really, it's fatty acid. So let me do OOH here before the chemistry people come and cut off my head. So let's take it, this is long chain with so many carbon in there. All right. Now what will happen is that this long chain fatty acid like this is not really useful to the cell. So it needs to be broken down into something we call acetyl-CoA. Okay, acetyl-CoA. Okay, all right, acetyl-CoA. Okay, this acetyl-CoA like that. Okay, and this acetyl-CoA is very useful. In fact, it can be used to build so many substances. Okay, so you break this long chain here, all right, into what? Into acetyl-CoA. And this breaking of this long chain, in fact, it's oxidation. Okay, is by, okay, so let me go up and come back so that I can, okay, so breaking of what? So one, okay, so breaking of the what? Of the long chain fatty acid, okay, is done by what? Oxidation. Okay, oxidation. And I hope you know oxidation, addition of what? Oxygen. Is that not it? Okay, it's done by oxidation. And so they will use oxygen. Okay, this organism is going to use oxygen. Okay, all right, it's going to use oxygen. So you can see even from my diagram here, there is what? There is a crystalline core. All right, good. So there's this crystalline core, all right, crystalline of what? Oxidase. Okay, good. That's what we are calling, what we are referring to as the catalyst. Okay, good. So what will happen is that we have the word, we have this long chain fatty acid will be what? Will be broken down into two carbon, like this two carbon structure. Okay, called the what? Acetyl-CoA. And this acetyl-CoA is very useful because it can be used to what? To build a lot of molecules. Okay, all right, good. So the breaking of very long chain fatty acid. Okay, the breaking of very long chain fatty acid. Okay, very long chain fatty acid is called what? In fact, the technical word for it is called beta oxidation. Okay, and so during this oxidation process, okay, one of the byproduct is the what? Is the hydrogen peroxide. So one of the byproduct is the hydrogen peroxide. Okay, one of the byproduct is the what? Is the hydrogen peroxide. So hydrogen peroxide will be released as a result of what? During the breakdown of this long chain fatty acid into what? Into acetyl, okay, acetyl-CoA, all right, into acetyl-CoA, all right, great, all right. So during that process, we will have hydrogen peroxide being released as what? As a byproduct. Okay. Now, another thing that can also happen is that sometimes we have this long chain fatty acid like this. Let's take it this way. This long chain fatty acid but it is like this. It goes this way. You know, you guys are doing organic chem. Okay, so you can see that when I do like this, it means that, okay, so this with branches. Okay, let's say when I do this, okay, this skeletal form of organic structure, it means these are branches, right? Okay. All right. A scientific term for this branch, we call them phenol. Okay, something like this. Let me see there. All right, like this, something like this. Phenol, like that. Okay, those branches. Okay. All right, great. So this branch here like this. This branch. Okay, these are branch word. These are branch. Okay, these are. So we have the branch form like this one, like this. Okay, so you have, this is quite long as well, but it has what? It has branches. So what will happen is that now when it gets into the peroxide peroxisome, the peroxisome will, when it is branched like that, it is not useful. So the peroxisome will use its enzymes, okay, Peroxisome will use its enzymes, and then we'll cut off this. Okay, we'll cut off this ones. We'll cut off those branches in it, all these branches to remove them. So to use the branch what? The branch fatty acid, it will remove the branch fatty acid, okay, and change it into what? Into long fatty acid like this one. That one is called alpha oxidation. Okay, so we have two types of what? Oxidation in the peroxisome. We have the beta oxidation, whereby the long fatty acid is broken down into acetyl-CoA, which makes it, this acetyl-CoA is very useful precursor for a lot of molecular processes. Then we have another, we have what? We have another one whereby a branch, okay, branch, okay, so that is the, so let me write that one here. So we have the, so let's say, okay, let's say we have the branch. Take this one out like that. Okay. So we have another one whereby we have what? Branch, okay, branch fatty acids are what? Are converted into very long word. Okay, there's no need for me to write all that. So very long chain fatty acid. So we'll convert this one, okay, the one here into this long one here. All right. So that one is called alpha oxidation. And during that process, hydrogen peroxide will be released as well. And as I said, hydrogen peroxide is what? It's a reactive oxygen, okay? And when it actually leaves the cell, that is dangerous. When it leaves the cell, it can destroy our DNA, can destroy our phospholipid, can destroy some proteins. And so, as being the hydrogen peroxide being the byproduct of what? Of the oxidations, okay, what happens is that we won't allow it to leave. Okay, all right. So after this oxidations, this hydrogen peroxide, which is the byproduct, will be what? Will be catalyzed or will be broken down, all right, by the what? By the catalyst, okay, into water and what? And oxygen. And you can see this is O2, and O2 is not a reactive what? Oxygen. O2 is not reactive. O2 is normal oxygen. So which means that we are fine to go. All right. So which means I've given you three functions of what peroxisomes here. I've talked about the breaking down using an enzyme called catalyst, and then undergoing beta oxidation. That's the changing of the word of the long chain, very long chain fatty acid into the acetyl-CoA. Then we also talked about what? We also talked about the branch chain, okay, converting branch chain what? Branch, okay. Branch chain fatty acids, okay. All right, branch chain fatty acids are converted into what? Are converted into very long chain fatty acid. And that one is called what? That one is called alpha oxidation. So we talk about three functions here. Okay, beautiful. Now, another thing is that they can also, so this one, all these points that we've talked about, these are under what? Under, that's the lipid metabolism. That's the breaking down of what? Of lipids. Okay, that's lipid metabolism. Okay, we can put these two under lipid metabolism. Now there is another lipid. In fact, there is this special lipid. Okay, let me duplicate this slide. Okay, let me duplicate this one. All right, so let's see, this ones can go now. Okay, great. All right, okay, all right, so we can go now. All right, now there's another very important lipid. Okay, this lipid is synthesized, okay, this lipid is synthesized in the, okay, so lipid, there's a special lipid, okay? Special lipid, okay? Lipid synthesized, synthesized by the word, by the peroxisome, okay? Peroxisome. All right, and this lipid is called what? It's called plasmalogen. Plasmalogen. Okay, and do you know what is this plasmalogen used for? You see, we will come to a certain structure very soon. This plasmalogen, okay, is what? Okay, so which called plasmalogen, and it is used to what? It is which is used, okay, for the formation, okay, of the myelin sheath in the nerves. Okay, we shall talk about nerve cells today, okay, when it comes to specialized cells, okay, but this particular lipid called plasmalogen, okay, this plasmalogen, okay, in fact there are a lot of them, plasmalogens, okay, great. There are number these plasmalogens, okay, what are they? So they are, okay, are used for the formation of what? Of myelin sheet in the nerves. And this myelin sheet, remember, they help in the transmission of what? Of nerve impulses, okay? So it is the work of the word. This particular lipid here, that the plasmalogens are synthesized in the what? In the peroxisomes, okay? They are synthesized in the peroxisomes, all right? Good. And then another thing that they can also do, okay, maybe I'll add this last one, okay, all right, they can also involve in what? Detoxification process, okay, they can also involve in detoxification process. So for instance, certain plants, okay, certain plants, and then and even in our liver cells, okay, in our liver cells. So okay, in some cells, so in some plants, okay, in some plants, all right, in plants, okay, all right, in some plants during photo, okay, all right, they are involved in something we call photo in some plants. They are involved in photo respiration. Okay, I will explain it very soon. Okay, so when you talk of photo respiration these days, now when it comes to photo respiration, what happens is that you know, ideally we expect that the plan should carry out when there is carbon dioxide presence. The
Hello,
plant
sir.
will have. Hello, please.
Yeah, in the second point, in
They
some plants, they are involved during in.
they are involved in okay, they are involved in, okay, let me put it that way. Okay, great. Thanks. So they are involved in photo respiration. So what is photo respiration at all? Like we have plants, you see, we expect that when carbon dioxide is high, the plant is going to absorb the carbon dioxide and carry out photosynthesis. That's what we expect. But occasionally, you will see that for certain plants, which are, we call them those C3 plant, like our plant we have here in southern part of Ghana, most of the plant, this C3 plant, what happens is that when temperature is too high, okay, when temperature is too high, okay, when temperature is too high within the leaves, instead of carbon dioxide to be picked up by the photosynthetic tissues or by the photosynthetic pigment, it will rather pick up oxygen. instead of what? Carbon dioxide. So to rather, it will start taking oxygen and respiring and giving out carbon dioxide instead. Okay, and this happened in plants, we call them C3 plants. Okay, most of the plants you see around are C3 plants. C4 plants are maize, millet, those cereals. Okay, those ones C3 plants. That's why you can see C3 plants. They do very well in hot dry areas. See, when you plant maize in northern Ghana, they do very well there. Okay, because they are not C3 plants. C3 plant in very hot condition, instead of them to undergo photosynthesis, they will undergo a mechanism we call photorespiration, whereby they rather take in oxygen and give out CO2 instead. Okay, and when that happens, it happens when the temperature is very, very high during the daytime as well. So we call that one photorespiration. So in some plants, they are involved in what? Photorespiration. All right, good. And then in us, we human, in our liver cells, they are there. Okay, and if they are going to be in our liver cells, then what are they going to do there? Okay, in the liver cells, they are also involved in what? In our liver cells, they are also going to involve in detoxification. So in the liver cells, so in the liver cells, so they are what? They are involved in detoxification. All right, good. Okay, so I think I've talked about almost 5 or 6 functions now. So let's do a quick recap of the perisosome. So it's very, very, very important. Okay, so you see, remember, okay, so if you come here, remember I said that they undergo more or less the lipid metabolism. And lipid metabolism, we talk about 2 main things. We talk about the beta oxidation and alpha oxidation. All right, and as a result of that, hydrogen peroxide will be released, and this hydrogen peroxide is harmful. It is reactive oxygen. When it gets out of our cell, it's a recipe for disaster. So it will be broken down by the enzyme called what? Catalase. Okay, good. Now, and then during the beta oxidation, we say that these two carbon molecule, which is the word, the acetyl, okay, acetyl-CoA will be produced. And this acetyl-CoA can be used for manufacturing so many other lipids in the cell. Okay, once the beta oxidation is carried out, so many other substances can come out of it. Okay, they can use it to build bile acid, other lipids, other useful lipids that we need in the cell. They can use this acetyl-CoA to build them up. Okay, great. And then, okay, so, and then we also talked about a particular lipid that is being synthesized, which is the plasmalogen. And plasmalogen is very, very important because they use it for the synthesis of myelin sheets. Okay, so the myelin sheet, that when we come to the specialized cells, we'll talk about it. Okay, the myelin sheet, they are around the nerve cells, okay, and help to speed up nerve impulses. And I said that in some plants, okay, we have the perososomes, okay, the perososomes. All right, the perososomes are involved in what? They are involved in, okay, the perososomes are involved in photorespiration, whereby they take in oxygen and give a carbon dioxide instead. So this is more or less a reverse of what? Photosynthesis. And it is bad because it causes the plant to lose carbon. Okay, all right. Instead of carbon being used for photosynthesis, it will not bind to the carbon dioxide. It will rather consume oxygen during very high temperature and give out carbon dioxide. Okay, and in the liver cells, okay, they are involved in detoxification. I think we talked about detoxification in our previous class, okay, whereby they break toxic substances down. Okay, all right, good. Okay, all right. So that is that for this one. Okay, great. So that is that for and it's very, very important again. In fact, there are a lot of diseases that if somebody perososomes are not functioning, it can actually cause. Okay, there is this popular one we call it Zwinga. Okay, this popular one. Okay, let's see. Okay, let's see. Do you even need this one? Okay, once I've mentioned, let me talk about it. Okay, so there's this popular one. Okay, yeah. It's the Winger syndrome. Let me double check this one for you. Zwiga. Let me double check. Let me double check this one. Let me double check this one for you. It's a disease. It's quite a very terrible disease. Okay. 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 Yeah, Zwiga syndrome. That is Zwiga. Okay. So let me bring it here. Zwiga syndrome. Okay, so the first one that I wrote is actually correct. Okay, so yeah, it's Zwiga syndrome. Okay, it's a very bad disease. In fact, it has so many disorders. This one is a genetic disorder. Okay, but it's a disorder when the what is going to bring the long chain fatty acid, they are not working. Okay, see, if you look at my diagram here, have you seen this one? You see, there is a channel here that is going to bring the long chain fatty acid. So in case there is a problem with this channel, the ABCD1 channel, when there's a problem with it, then it means that they can't really bring the very long chain fatty acid into the perisosome for the oxidation to take place. And when it happens like that, it leads to accumulation of the very long chain fatty acid in the blood. Okay, and that causes the Zwiga's syndrome. So it can accumulate in several places in our body, sometimes even on the eye and stuff like that. So people can't see very well. Okay, all right. There's even this disease, we call it ataxia. Okay, it leads to a lot of disorder. There's one too, we call it retinitis pigmentosa. Like the person's eye will be there, the eye will be like that. This is just by the way, so that the eye will be here like this, the eye here like that. The people will be here, and then right in front of his eye, he can't. This side of his eyes, okay. When on his eyes, he can't see this side. Okay, it's only straight. It's only this side that you can acquire because fat accumulates on this side of the eye. So the people, like if this is the people, like it means some parts here are covered. Okay, all right, and then we call that one retinitis pigmentosa. This is just by the way, so don't worry. It's so much. Okay, so even though it is a very tiny little bit of organelle, but when they are not working, they wreak so much havoc in the cell. Okay, all right, good. So we are done with almost all the membranous organelles. Okay, let's talk about non-membranous membranous organelles. Okay, and the almighty one we're going to talk about is the what? Is the ribosome. Ribosome. Okay, the ribosome. So let me pick ribosome picture online for you. Let me pick ribosome picture online for you. All right, so let's see. I can get ribosome picture. Okay, ribosome. Okay, yep. Let's see. Okay, yeah. So let me take this one. Okay, I'll take this one. All right, and then okay, so let me take this one here. All right, so okay, good. That's a very big one here, very big ribosome. Okay, good. That is it here. Let me put this one here. Then let me bring another one that will also help me to talk about what I want to talk about. Okay, great. Okay, great. I've also gotten some nice one here. So let me copy that and then bring it here. Okay, so this is that. All right, nice. Okay, great. So these are my ribosomes. Okay, great. Okay, great. So these are ribosomes. Okay, great. Let me crop some parts off so that great, beautiful. Okay, let me go take this one off here. All right, great. All right, so this is my ribosome. Okay, these two. Okay. All right. Great. So these are my ribosomes. Okay. Let me. Do you know what? Let me put these two on one side so that. Okay. All right. All right. So let's see something like this. Great. Let's see something like this, like that. Okay. All right. Good. Okay. So now I'm going to talk about ribosome. Okay. So this ribosome, I think the previous slide gave you a head start on it. A ribosome consists of what? So when it is what we know it is what? It's non-membranous. It is what? Non-membranous. Okay. Membranous. Okay. All right. It's non-membranous. All right. So it is non-membranous. That's the first point. The second point is that it is what? It consists of what? It consists of ribosome RNA, okay, which we call it what? RNA, okay, and what? And protein, and proteins. Now, the composition of the proteins, the composition, so the composition, okay, the compositions, okay, the compositions differ among what? Among different what? Organisms. Okay. All right. So for those of us, for humans, okay, it's about what? It's about what? 50% protein, and what? And 50% RNA. Okay. Okay, for humans. Okay, but it differs for other organisms. Okay. All right. Great. Okay. So. All right. Great. All right. And then what will this one? So they come together. Okay, to form. And then where is this assemblage? Okay. Remember I say that it is where it is assembled. It is assembled in the part of the cell. So it's assembled. Okay. Assembled in what? In the nucleolus. Okay, in the nucleolus. If you remember, okay, the last time we told about it, it's assembled in the nucleolus. Okay, great. Now, after the assemblage, what do we get? You see, we have two main sections or two main subunits. Okay, let me even take this one off a little bit. Okay, so we have two main subunits like that. And this subunits that you can see from here. So we have large subunits. Okay, so there are two subunits. Okay, they are what? There are two subunits. Okay, subunits. Okay, all right. And these two subunits, we have the what? We have the large subunits, and then we have what? Okay, large and small. Okay, let me put it this way. Right, great. And as you can see here, look at that. So you see that you can see the small one at the bottom here, and then we can see the large one at the top here. All right, good. So now these two together, they are not always together. Okay, in fact, they are not always together. All right, they come together, okay, during protein synthesis, but they can, okay, and these two, they are of different work. They are of different, let me put it this way. They are of different units of measurement. So let's take it, there is a way we measure this one. We measure using something we measure the ribosome using something we call sedimentation coefficient. Okay, sedimentation coefficient, and then the sedimentation coefficient was named after a scientist we call it was Silverbeck. All right, good. This one. So this scientist that they named it after him. Okay, so we call, so the segmentation, as you can see from here, is more like the rate at which it settles, the rate at which it sediments. Okay, so that's the segmentation coefficient. That's what we use it to measure. Now, these ribosomes here, the large one, okay, I think I can duplicate my slides. Okay, let me duplicate my slide here. Okay, all right, good. So, okay, so now these points, we've made these points already. All right, great. Now, these two subunits, we have measurements. Okay, so we have the large subunit and small subunits. Okay, and then we measure them using something we call segmentation coefficient. Okay, and the unit is called Sievertberg. Okay, it was named after the scientist who discovered it. All right, and so the large subunit, okay, the large subunit. Okay, so here, let me even be a little bit specific. So for prokaryotic cell, prokaryotic cells, okay, the ribosome there, the small subunit is what is so the for eukaryotic cell is 30. Okay, it's 30. All right, 30s, 30 Sievertberg, and then the large one is what is 50. The large subunit unit is equal to 50x. All right, so you see, so as I say, these two subunits, they are assembled, all of them are assembled at the nucleolus. Now, when they assemble and then they come to the cytoplasm. Okay. Now, it's not always that these subunits they are always together. No. Okay. In fact, the two of them must come together to form the ribosome. All right. So what happens is that most of the time they are not together. They come together when protein need to be synthesized. That's when they usually come together for the synthesis of what? For the synthesis of protein. All right. Now, so please just a minute. For the synthesis of what? For the synthesis of what? Of protein. So we have the small subunit is what? Is 30s, and then the large subunit is what? Is a 50s. But do you know what? When they come together as this, okay, when they come together, this one. So here, this one for prokaryotic cell, this large one here is what? Is 50s. The small one here is what? Is 30s. You see, when they come together and you measure their sedimentation, like when they are together, so together, now listen, when they come together, it is where it is 70s. We don't add them. So when these ribosomes are together and then you calculate their sedimentation, the rate at which they settle as together, it is equal to what? It is equal to 70s. So here it's not addition of this and that. No, this is a chemical, it's like a physical process. Okay, the rate at which it's going to settle. So it's just like maybe I'm buying something and then they told me that, oh, the sachet water, one is let's say 40 pesos, but I can give you 3 for 1 cedi. Okay, so when they are together, when they are individually, the small one is 70s, the large one is 50s. Okay, but when they come together, when they are together and you calculate their sedimentation coefficient, okay, it is where it is 70s. All right, so please, so remember that prokaryotic cells, they have 70s ribosomes, and this 70s ribosomes is similar. It's what? It is similar. It is what? It is similar in structure, okay, similar in structure to that of what? That of the ribosomes in the mitochondria and chloroplast. Okay, chloroplast. All right. Okay, so they are similar. Okay. Also, so this one is even confirming the word the endosymbiotic theory. Okay. All right. So together they are what in prokaryotic cell 30 and what and 50. But when they are together and you measure it, it is what 70s. Okay, all right. And I said this 70s ribosome is similar to that of what? It's similar to the mitochondria ribosome as well as the chloroplast as well. They are not the same, but they are similar. Okay, all right, great. Now let's go to the eukaryotic word. For eukaryotic, let's see the eukaryotic. Okay, for the eukaryotic, eukaryotic. Okay, so for the eukaryotic cells, okay, for the eukaryotic cells, what is going to happen here? They have what? They have the subunit. The small subunit is what? Is 40 S, and then the large one equals 60 S. Okay, 60 S, and together when they are together, okay, when they are together, it's what? It's equal to 80 S. Okay, all right. So we have what? 80 S ribosomes. So you can see clearly here that you see that is why if you are developing antibiotics, you can easily target. There are a lot of antibiotics that tend to target what? The ribosomes. Okay, there are a lot of antibiotics, plenty of them. They target the ribosome. Why? Because the ribosomes in our cells and the ribosomes in bacterial cells, they are different. So these discrepancies in the word, in the composition of the ribosome, okay, they are highly exploited in the production of what? Antibiotics. Okay, so let me put that point down because that's a very important point. Okay, so all right, so differences, the differences, okay, the differences in the composition, okay, of the ribosome, okay, ribosomes of what? Of proca, okay, let me be specific, of bacteria and eukaryotic cells, okay, eukaryotes are highly exploited for the production of what? Production of antibiotics. Okay, I hope you know antibiotics. Okay, we know antibiotics, right? Okay, do you know antibiotics? What are antibiotics? What's antibiotics, please? Yes. What's antibiotics? Yeah.
They are drugs. They are drugs that are made to fight bacteria. They are drugs that are mainly used to fight infections and bacteria.
Okay, that are mainly used to fight infections in bacteria. Oh, okay, that's good. Okay, so that is it, basically. Okay, so there is this classic one and stuff like that. So here, so as I just said, you see, if you are taking drug, the drug should be able to kill the parasite, but it shouldn't kill you. So you see, it is good that they have 70. You see, their ribosomes are different from our ribosomes. So I can design drugs that can target 50S ribosome. So you see, I don't have 50S ribosome. Have you seen it? I don't have 50S ribosome. All right. And so I can develop drug that can target 30S ribosome. I don't have 30S ribosome. So which means that drug wouldn't affect me. Okay. All right, good. I don't know whether you there is this eye drop. Okay, this eye drop that we normally take. If you check this eye drop, you see this antibiotic in it. I buy it quite a lot. So gentamicin. Okay, gentamicin. Okay, good. This eye drop like that, it targets the what? It targets the ribosomes of the bacteria. Okay, all right. This particular antibiotic. Okay, you don't need to put this one in your head, please. I'm just it's something you come across it. That is why I want you to know of it. And then there is this group of antibiotic, we call them tetracycline. Okay, even our almighty streptomycin. Okay, they all streptomycin. Almighty, the one we've been taking, streptomycin, right? Streptomycin, almighty streptomycin. They all target the word, the ribosome. And then the classic word, tetracycline. Tetracycline is also tetracycline. Okay.
Yeah.
All these. Yeah, let's do this.
What about chlorophenicol? Chlorophenicol. Anytime. Yes, when you develop Apollo, and you put the eye drop.
Yes. It's also it's targeted. It also targets the chlorophenicol. Yeah. You're right. Chlorophenicol also targeted. That one it even targets the it's not even directly, yes, but what happens that during this protein synthesis, okay, it blocks like here, like you see, I've shown that protein synthesis. So chlorophenicol, what it does is it blocks the formation of this bond. You see, when they bring the amino acid here, okay, there should be a bond that will form between the amino acid. So that bond is called peptide bond. Okay, so let's say if you look at my diagram here, okay, that's why I deliberately brought this one. You see, what the chlorophenicol does is that it blocks, okay, it blocks the formation of the peptide bond during the protein synthesis. All right, great. But it is still associated with the ribosome, so that's correct. Okay, good. So, in fact, there are so many antibiotics. Yes.
Hello,
please.
Doc. Please, the eukaryotic cell side, when you put the two together, I don't know the number that I'm seeing there. I don't know what.
No, we don't add demo. You can see that this one when you add it, it is 80. No.
So
It's not. No. It's not that you don't do normal addition. So watch out. In exam, I can trick you. Trick you. Watch out. Good. Why God has to do something to you. Okay. So hey, our ladies to their spiritual, God reveal things to them. Okay, good. So here, when you add the two, when they are together, it's not 80. Here, when they are together, it's not 100. When they are together, it is 80. This one, when they are together, it is what? It is 70. All right. So these differences in the structural composition of the ribosome is exploited for production of antibiotics. You see, as I said earlier on, when you are hello.
Yeah. So what if they are together and they are exactly 80? What happens?
It won't be exactly 80.
There will be no circumstances where we'll get exactly 80.
Not at all. When they are together, this is 8. The 80 is not just addition of this one. We call something sedimentation coefficient. So when they are together and you run, when you say, you remember sedimentation, when something is settling down, okay? When they are together, the rate at which they settle down in a solution is so it's not that's addition they are talking about, you know. It's not that they are adding some chemical. No. It's the rate at which they will settle down. Let me draw something for you to see. So like this. This is experimentally determined for over more than 100 years. Okay, so here, listen. So when you have this one, so this one alone, the rate at which is going to settle down here in the solution. Okay, when you have the small subunit, the rate at which will settle down, and then the large one, let's say here, the rate at which it will settle down. And when the two are together, this and that are together. Okay, the rate at which they settle down is 70s. That's the sedimentation coefficient, and the unit is called Sievertbeck. Okay, so when this one is alone, okay, it will settle down at 50. When this one is alone, 30. But when they are together, the rate they settle down is 70. So it's not that there is a special addition of you are increasing volume of something. No. It's the rate at which they settle down within the solution. That's what it is measured at 70 S ribosome. Did you understand what I'm saying? It's not that there's a chemical, one volume is 30. It's no volume. What is here? It's no volume measurement. That you say that I fetch water 70, I fetch water 50, I fetch another one, and then when you add the volume is 80. No. This is sediment, the rate at which they settle. Okay, all right, great. Okay, and it has been, these figures are empirically determined. So which means that it was something that has been done in several labs for several years. Okay, and it has been established at 70 years when they are together. Okay, thank you. So the differences in the compositions, okay, this differences in the composition is what? It's the difference in the composition is what is highly exploited, all right, because if bacteria is having 70S or specifically has 30S and 50S ribosomes, and then the eukaryotic cells are having their what? Are having 40 and 60. They are different. And so I can design antibiotics that will target 50S ribosomes. Okay, all right. And so with 50S ribosomes, you can see clearly that, okay, it's when I take that drug, it wouldn't affect me. Why? Because I don't have 50S ribosome in me. I have 40-60. If the person design ribosome that is going to target, let's say ribosome that is going to target, let's say 30S, okay, the 30S ribosome will not do anything to my body. Why? Because I don't have 30S what? I don't have 30S ribosome. Okay, so you see that is why a lot of antibiotics tend to exploit the what? The ribosome. Okay, I remember another structure we also talked about previously is the what? Is a peptidoglycan, because bacteria have peptidoglycan, but we eukaryotic, we don't have peptidoglycan. So that one can also be exploited. For so penicillin, our almighty penicillin, it targets the what? Peptidoglycan. Okay, and then gentamicin, streptomycin, tetracycline, and stuff like that. Okay, they all there are so many of antibiotics. In fact, most of them, far majority of them, they target the what? They target the ribosomes. Okay, because the ribosomes, when they target the ribosome, they will stop protein synthesis. Okay. They will stop the protein synthesis. And once organism can't make protein, that is the end of it. Okay. Once you can't make protein, because you need protein to produce your enzymes, you need protein to produce the hormones, and you need protein for a lot of metabolic activities in the cell. So if the proteins are not being made, then of course that is the end. Okay, all right, good. So this is highly exploited, okay, good, for production of antibiotics. And the ones that are, in fact, there are a lot of antibiotics we can get. If we say we are going to mention them, we will not leave here. Plenty of them, okay, erythromycin, azithromycin. Azithromycin is quite popular. I think it's quite popular. We take azithromycin quite a lot. Okay, streptomycin is also quite popular now. Penicillin is no longer popular because the bacteria develop resistance to it. Okay, so it's not so much popular. Right, good. So
amoxicillin.
Yes. Oh, almighty amoxicillin. Yeah, almighty amoxicillin. Yes, please.
In some instances, you take some antibiotics and you have a heartburn.
Yes.
What would be the cost?
Please, I wouldn't know. That one I wouldn't know. Every drug has its own side effects. Okay. I wouldn't know specific antibiotic that you take that will cause that. But definitely once it's a drug in your system, I wouldn't know which one you picked. Okay. If the heartburns are very too much, please report it to the doctor. Okay. I wouldn't know because there are many. These drugs, there are many. And I'm not into maybe they're like the specific mechanisms of the drug. I only know the surface stuff, okay, of the drugs as geneticists, okay. All right, good. Okay, so these ribosomes, okay, the structural differences in the ribosomes are highly exploited for the production of antibiotics, and then we've mentioned quite a number of them, okay, gentamicin, streptomycin, tetracycline, all these ones that I've written here, they target the ribosomes, okay. All right, good. So then so these ribosomes are very, very important. Okay, good. Now remember that we have two main types of even within the cell, we can talk of what two main types of ribosomes. Let me duplicate my slide again. Okay, so I can delete the top one here. Okay, all right, good. So within the cell, we can also talk about what? We can also talk about two main types of ribosomes. See that we have ribosomes, okay, ribosomes that are attached, okay, to attached to the rough okay, so more or less, and then we have the what? We have free, and then we have what? And we have ribosomes that are what? That's a free. Then we have the word free floating ribosomes. Okay, so let me just call this one. Okay, so free floating ribosomes. All right, good. So research has shown that those ribosomes, those attached ribosomes, attached. Okay, so attached ribosomes, okay, make proteins, okay, make proteins that are destined to become part of the lysosome, or cell membrane, cell membrane, or secreted out of the cell. Okay, all right. Then those free floating ribosomes. Okay, so let me take this one up. Let's already talk about this. Okay. Then free floating ribosomes. Okay. Then free floating ribosomes. Okay, so those ribosomes floating in the cytoplasm, okay, free floating ribosomes, okay, produce or make, okay, produce proteins that are used in the cell. Proteins that are used in the cell. Okay, so we have the free floating ribosomes. They produce what? Proteins that are used in the cell. All right, great. So within the cell, okay, so within the cytoplasm like that, the proteins that are used within the cytoplasm, okay, of the cell, that are coming from the free floating one, while the attached ones, they are either going to be destined to become part of the lysosome or the cell membrane, or will be secreted out of the cell. All right, good. Now, so now that we've talked about this in a little bit detail, let's see this ribosome here. All right, good. Now, if you look at this ribosome here, all right, if you look at this ribosome, you can see that what is the ribosome going to do? You see, we have this large subunit and then the small subunit. The large subunit, remember that I said the last time when we talked, I said that during protein synthesis, there is this messenger RNA. Okay, so let me make a little bit of sketch. Okay, so let me take the marker here. So during protein synthesis, we have the what? We have the messenger. So this is the nucleus here. Okay, so the nucleus. Okay, so let me draw it. Well, so this is my nucleus. You know what? Since I have this is somewhere. Nuclear envelope. Nuclear envelope. Oh, I don't have the nuclear envelope here. Okay, no problem. So like that. Okay, like that, you see. I'm delaying, let me get this image. Okay, so nuclear envelope, that will make life easy. Fast. Okay, so let's see, nuclear envelope. Okay. Okay, now if I bring this my nucleus here, so if I bring this my nucleus here, okay, okay, good. All right, so let's say I've brought this one here. Now you can see clearly that in this there's this nuclear pore. Okay, these are the holes. So it is this hole that all the instruction for the protein to be, the type of protein to be manufactured will come from the DNA inside the nucleus. All right. So what is going to happen is this. In the case of eukaryotic cells, okay, whereby the DNA is confined in the nucleus, the DNA is too big to go through this nuclear pore. Okay, so this nuclear pore here, all right, the DNA is too big to go through it. Okay, so we have the word. So the DNA is too big to go through the nuclear pore. So what is going to happen? What is going to happen is that the information for the specific protein to be synthesized will be copied into a type of what? RNA. And that RNA is called messenger RNA. Okay, all right, so here, so the information from here, from this will be copied, okay, so information, okay, on the what, that genetic information, okay, so let me make it a little bit specific. The genetic what information, okay, the genetic information on the DNA, okay, will be what? Will be copied, all right, will be copied, okay, will be copied onto the word, will be copied onto the messenger RNA, okay, will be copied onto the messenger RNA, all right. That process is called transcription. Transcription. Okay. All right. Okay, great. So that process is called what? Transcription. So the information will be copied onto messenger RNA, okay, which we call mRNA. The process, okay, the process is called what? The process, okay, the process is called transcription. The process is called what? The process is called, okay, it's called transcription. All right, great. Okay, the process is called transcription. All right. So this messenger RNA will then what? will then leave here. Let me draw. Well, then this messenger RNA will then leave here. So the messenger RNA, so let's say this is my DNA, they've copied information onto the word messenger RNA like this. Okay, so mRNA like that. So mRNA is carrying the genetic information like this. Okay, so this mRNA, okay, like this, will now pass through this hole here, like that. And as it is coming with genetic code. Okay, and this special genetic code, they've been arranged in triplets. Okay, they have been arranged in triplets. Okay, so this triplet, let's say the triplets, let's say maybe let's say, let me call it maybe D. Okay, let me use the specific one. So let's say U, A, C. There are 3 alphabets. Okay, there. So we'll come to it. Okay, don't worry, we shall come to it. So let's say U, A, C. So this UAC, this triplet of alphabet, we even have something like UCC. Okay, we have what CAU. We can get a lot of them. Okay, great. So this, they will be carrying these information, triplet of information, and this triplet of information that they are carrying from the DNA. Okay, that they've been copied it onto the word messenger RNA. So on the DNA, we call it, so the DNA will have something called genetic code. Okay, and when the genetic code is copied onto the messenger RNA, on the messenger RNA, we don't call it code, we call it what? Codon. Okay, codon. So this triplet codons. Okay, so these triplets like U, A, C.
Hello there.
Yes, please.
So with the DNA, isn't it actually in a static form or it's in a solid form? Because with the mRNA, which can move out of through the nuclear pores, but with the DNA, it can move out. So it's in a static form that can't move, or in a solid form.
The molecule is big. It's too big to go through this hole. The solid.
Okay.
The big molecule. The molecule is big. Okay, it's a big molecule. The molecule is too big to go through. And this one, messenger RNA is not big. Messenger RNA is very small. And besides, you see, it's not let's say your DNA is like this. Your DNA is very long, like this, like that, like that. Okay, let's see, this. Okay. All right, something like this. All right, if protein is going to be made, it's not the whole of this. Let's take it this way, that Angela has given birth to a bouncing baby boy, and the bouncing baby boy needs to be fed with milk. So it's not the whole of the DNA that is responsible for milk production. It is just small part. So here, maybe this part of the DNA is where the milk production is responsible. This side is what is responsible for production of milk. Not every part. Okay, have you seen it? It's not every part of the DNA that produces milk. All right. So this side here, it is the messenger RNA. So once, and if it's not even in every cell has the same DNA, but it's not every part of the DNA is activated. So in the case of, let's say, Angie, bouncing baby boy for Angie, and then the milk that's needed. So it is only the cells in the breast, the lactocyte. The lactocyte in the breast will be activated. There's a specific part of the lactocyte, the DNA in the lactocyte of the breast, because not every cell in the human body will be producing milk, right? So it is the lactocyte cells in the breast that within that cells, their DNA, some parts of the DNA here. So just here, that the information here will be, the DNA will open and information here will be copied onto the, so you see that this long DNA, it is just a small one that will be copied. And even when it's copied, it's not double-stranded. It's a single-stranded. Have you seen it? So it's very small. So this small thing that will come out. Okay, let me use different colors so that. Okay, so it will be like this small thing here. Okay, this is the messenger RNA. Okay, like this small one from here that will come. So let's say this is my mRNA. Okay, all right. Okay, because the baby has been born, we need milk. So just the side, maybe just the side of the DNA responsible for milk production. This part will leave. Okay, will be copied onto the word messenger RNA. The messenger RNA will leave like that. And when it comes, the small ribosomal RNA will anchor it. So you can see that, look at it. You see the small ribosomal RNA, there's a kink here. Have you seen it? Okay, so it will anchor it, to capture it. Okay, then they will start translating. So if you look here like that, so you see this is the ribosome, the messenger RNA here that has come here, and each of the messenger RNA is carrying what? Triplets of what? Of bases. And this triplets of what? Of the bases here is what we call what? Codon. But on the DNA, we call it what? Code. And then it will be copied onto messenger RNA, and we call it codon. So they will go this triplet, another triplet. So it goes on and on and on. Another triplet. So 3, 3, 3, 3, 3, like that. Okay, 3 like that. Okay, so it goes on and on. So these triplets of what? These triplets of the codons is what will be translated. So you can see here that now we have something called transfer RNA. So transfer RNA, based on this codon, will go and pick specific amino acid and bring it. So this is the transfer RNA, okay? All right. So the transfer RNA will come here, okay? We'll based on this sequence here, we'll pick, so like this sequence, like the UCC like this, it picks an amino acid we call serine. Okay, I don't know the amino acid, but I know this one, you know, because of UCC, you can't forget it. The big amino acid called serine. So when this codon here is UCC, the amino acid that will be picked, here, that amino acid is called serine. Okay. So based on the messenger RNA that will come, the sequence of the messenger RNA, then the transfer RNA will look at it and go and pick specific amino acid. So if you look at my diagram here, you can see transfer RNA is picking specific amino acid, come and then bring it here and then add it to then form a chain of what? Amino acid, which we call peptide chain. Okay. All right. So when it comes like that, so they'll put one amino acid here, another amino acid here, and then there will be a bond. This bond is called peptide bond. Peptide bond. So it will continue like that until there's one codon, we call it stop codon. So when they see stop codon, then it will stop. And the protein synthesis will stop. Okay. So this is basically, so that is why we say that ribosome, okay, the ribosome is what? Is the site of what? Protein synthesis. Okay. Let me duplicate this slide one more time and then okay, so this one can go now. Okay, then let me tidy up the surface here a little bit. Okay, so what is going to happen is that so here we know that the messenger, so mRNA, okay, will come out of what? Come out of the, so this, we are talking about, this one is specifically for eukaryotes. Eukaryotes, okay, eukaryotes. Okay, so the mRNA will come out of the what? Out of the nucleus, okay, or will come out of the nucleus through the what? Through the pore. The pore, and then will be what? Will be anchored by the what? By the small subunit, small subunit of the what? Of the, will be anchored by the small subunit. Okay, will be anchored by the small subunit of the what? Of the ribosome. Okay, of the ribosome. Okay, great. And remember, this one, the messenger RNA coming is carrying what? That genetic information we call codons. Okay, codons. All right, okay, ribosome. Okay, so when you get there, then the codons, okay, will be what? Translated into specific word, specific amino acid, okay, to form a protein chain. All right, good. So the one who is going to do the translation is a what? Is a T transfer RNA. So the transfer RNA will pick the specific amino acid and then we'll join like that together. Okay, so one amino acid and then the peptide bond will connect it and then eventually will form the protein. Okay, so that is why. So from here you can see clearly why ribosome is the word. It's regarded as the site for protein synthesis. Okay, please, any question for me? Any question for me before we proceed? Any question for me? So you can see that the cells that are actively involved in protein production, they are what? They have numerous ribosomes, okay? All right, and then they have well-developed what? Well-developed rough ER, and then also well-developed Golgi body, because the Golgi body is where the modification of the proteins will occur. All right, is any question for me? Any question for me? All right, so now that we are clear with this one of the site for protein synthesis. Now, do you know what? You see, apart from the fact that we have the ribosomes, okay, we have other structures in the cell too that also contain ribosomes. So we know the mitochondria, and what? And chloroplasts. Chloroplasts also have what? Ribosomes. All right, mitochondria and chloroplasts also have ribosome. And this one is confirming the word. This, okay, this is what? This is confirming the word, the endosymbiotic. If you could remember when we did the last endosymbiotic theory. All right. Okay, great. Enough of ribosomes. Okay, all right, good. Now let's go to another non-membranous organelle. Okay, another non-membranous organelle. So the another non-membranous organelle that we can talk about is what? Is what we call centriole. Okay, centriole is a very, very important non-membranous organelle. Now, this centriole, how is it like? Okay, in fact, okay, let me get a picture of it quickly for you. Centriole, let me get a picture of it. Okay, great. Okay, but then this one is nice. Okay, great. Copy it here and then let me bring it here. Okay, so let me bring this one here. All right, so this is no, I don't want it this way. Okay, no, so, all right, great. So let's see something like this. So that's all right, good. So we have this structure I have on my screen here. That's the centriole, okay? And this centriole is particularly important, okay? Centriole is one of the most important structures in the world, in the cell. Very, very, very important, okay? Very, 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 very important, all right? And then, so it is, well, obviously we know that it is what? It is non-membranous what? Organelle, okay? It is non-membranous organelle. Okay, so let's say, so first of all, non-membranous organelle. So it's not bounded by membranes. Okay, good. It's non-membranous organelle. Now you can see, if you look at my screen here, have you seen that? Look at the way they've been arranged. if you look at it, if you look at the way they've been arranged, you can see that no, these structures that are forming the word, these structures that are forming these centroles, okay, we call them microtubules. Okay, we call them what? Microtubules. Okay, so the microtubules, okay, so look at it. So triplets of what? So you can see that now look, let's count it in. So look at this one. So we have the triplets of microtubules, and if you count it and tell me what number will you get? Count it and tell me what number will you get? I want you to count it and tell me what number will you get?
3.
No, when you count, it's 3 here. But how many of the triplets?
9.
Good. So we have what? We have
9.
good.
Nine.
Excellent. Ah, my lady has said this, so that's correct. So triplets of what? So triplets of triplets of 9 triplets of what? Of microtubules. Okay. So these, each one is a microtubule. This structure, okay, is what? It's formed from what? 9 triplets of what? Microtubules. Okay. All right, good. That is forming it. Okay. And one may add, okay, so this triplets of microtubules that we have is a very nice, beautiful structure here. Good. And then you can see that there are 2 of them arranged well. Now these 2, okay, so we have 2 of them. Okay, so 2 of them, okay, 2 centroles, okay, centroles, all right, they lie where are lying perpendicular. They are lying where perpendicular. I hope you understand perpendicular. Okay, perpendicular to there lies 90 degrees. Okay, perpendicular to each other. Okay, each other. And when we have the 2 together, we call that one centrosome. So when you talk of centrosome is basically the 2 centroles that are lying perpendicular to each other. Please, are you with me? So the centrosome is not any special thing. It's just the 2 centroles that are lying perpendicular to each other. So when we have these 2 together collectively, okay, we call it what? We call it centroles. Okay, good. Now, what is this doing? Now, in as much as these structures that we are talking about is very, very important. Centroles are what? Centroles are what? Absent in plants and fungi. Okay, so in fungi, okay, fungi and plants, they lack centroles. So one of the differences that you can also add for difference between plants and what? Animal cells. Okay, now I normally ask funny questions. You see me asking questions like this, that write 2 differences between fungal cell and animal cell. All right, what will you say? Let's see if I ask that question in exam. What would you say if I ask you to write 2 differences between fungal cell and animal cell? What would you write?
Hello, Doc.
Yes, please.
With the animal cell, they lack chitin, and the fungal cell, they have chitin as their cell wall.
Good. Then the second one. Well done. All right, good. Well done. All right, so you can see the second one here. The second one, presence of central and absence of central. So you can see that the animal cell, they lack the cell wall. Okay, but the fungal cell has a cell wall, okay, consisting of what? Of chitin. Well done. Ooh, proud of you. Good. So you see, I normally ask questions that's why I have to take your time and make sure you understand whatever that I'm teaching, because the same way I teach is the same way I ask my questions too. So please make sure you understand it. Okay, great. So the centrals are absent in plants and in fungi. Okay, all right, great. Now, so what is the central? Why is this central important at all? In fact, these centrals, okay, the centrals, okay, they are what? The centrals are microtubules organizing organizing what? Center. Okay. They are microtubule organizing centers. See, they organize the microtubules, okay, for what? They organize the microtubules to form organizing centers, microtubule organizing centers, for what? For formation of spindle fibers. Okay, so here we know that the centrals are what the absence in plants, okay, plants and what fungi, but presence in what? Presence in animals and what? And prototysts, some prototysts, not all of them, some prototysts. Okay, so they are microtubules organizing centers, and they organize. So this is so during cell division, like mitosis and meiosis, okay, which I talked about in brief the last time. During the cell division, you see in animal cell and in some prototysts, there is this thing we call it what? So this what is here is the spindle fibers, and so what is going to the centrals? You can see central here, central there. So the centrals, during the cell division, these centrals too will divide, and then they will move to the opposite poles. Okay, so they will move to the opposite poles of the nucleus, and then that's where they will start the formation of the what? Of the spindle fibers. Okay, but as a matter of fact, fungi and then the what? And plant, fungal cells and plant cells, okay, they can do what? They can divide without the formation of spindle fibers. So which means that spindle fibers formation is not strict prerequisite in what for cell division, because some cells can divide without forming the what? Without forming the spindle fibers, and that's typical example is the what? Is the like without using the some can divide without even forming the spindle fibers. Okay, and then with some also, which means that some can also divide, okay, without the what? Without the central. Okay, some can divide without the central. So you can see plant cell can divide, they can even form the spindle fibers without fungi. Some fungi cell can form spindle fibers without the what? Without the central. Okay, because they don't have central and they still be able to form the spindle fibers and divide. Okay, all right, great. So but for the cells that tend to have them, like the animal cells and then some prototysts, okay, all right, the centrals, they initiate the what? The formation of the what? They organize the microtubules for what? For formation of spindle fibers during what? During nuclear division, nuclear what? Division. Okay, and when I talk about nuclear division, remember I'm referring to what? I'm referring to mitosis and what? Meiosis. All right, mitosis and meiosis. Okay, mitosis and meiosis. All right, good. So and then aside that too, apart from the formation of this one of the, okay, they can also what they can also get involved is that sometimes there is this structure, okay, that is the flagellum. You see the flagellum and then the cilium, okay, so the plural, that's it. They can also sometimes they form the anchor body. You see the basal, but there's something called basal body here. So let's take it this way. If I have an organism like this, let me see, I can draw. So let's say I have a, okay, so if let's say I have something, let's say let's take it this way, that organism like Euglena. Euglena looks something like this. Okay, so this Euglena has what? Has a flagellum. Okay, let's say like that. Okay, so this Euglena has a flagellum like this. Okay, now this flagellum, the basal body here, there's something called basal body here. So the basal body is basically, it is formed from what? The central. So they form the basal body from which, okay, they form the basal body here, okay, from which the flagellum is what? Is anchored, all right? So they form the basal body from which the flagellum is what? the flagellum, okay, or the cilia is what? It's a anchor. So we have the word, the basal, okay, body. All right, okay, so they form this one. So another thing we can add, apart from acting as the microtubule organizing center, we can also have what? We can also, let me put this one up a little bit. They can, so, okay, they can serve as what? So they also serve as what? Serve as the basal body to anchor the way the cilia and flagellum. All right, great. Okay, so the cilia and flagellum, like that, even from what you can see that they also made up of what? Microtubules. Okay, all right, good. So these are two main functions that we could talk about for cells that tend to have the what? For cells that tend to have the centrioles, okay, all right, the cells that tend to have the centrioles, one, they act as what? Microtubule organizing center for the formation of what? For the formation of the spindle fibers during nuclear division, okay, in mitosis and meiosis, and they also serve as the basal body to anchor the cilia and flagellum. Okay, all right, good. Okay, so I use the plural synonyms, flagella. Okay, all right, great. So we are done with this one. So the last one I'm going to talk about is the cytoskeleton. Okay, so as you are, let me take new slide and then good. So let's talk about the cytoskeleton. Cytoskeleton. Same. Okay, good. So when we talk of cytoskeleton, what is it? What is it at all? Now, when we talk of cytoskeleton, there are three main things we talk about. Okay. All right. So the cytoskeleton, we talk about three main things. What are we referring to? We are talking about the microtubule, which we just talked briefly about it. The microtubules, okay, and then the what? The microtubules, and then we have the intermediate, okay, filaments. All right, and then we have the what? And we have, and the last one is the what? Is the microfilament. Okay, we call it micro. Okay, microfilament. Into bracket. Also, it's also called what? Actin filament. All right. So these are the three structures, the three organelles. When we talk about the cytoskeleton, these are the three organelles that we are referring to. So the name cytoskeleton, obviously, which means that it's giving some sort of what? Rigidity, okay, some sort of stability. So in general, which means that it's like skeleton that. So it gives what? So cytoskeleton, it gives the cell some sort of what? Stability, okay, the cell and then the what? The organelles. So in the word, the organelles, some sort of what? Stability, okay, stability, okay, some sort of stability. All right, great. Okay, so we can talk about individual ones. Okay, so let's talk about this one briefly. I won't go into too much detail. Okay, so let me talk about this one briefly. So microtubules like that. Okay, as we saw it here. Let me just have a picture somewhere. Not really. We won't go into too much detail, so I don't think I have to bother your head that much. But then you can see that from what I did here, these rods are the microtubules, okay? And these microtubules, they are actually coming from protein. The protein is called tubulin, okay? Tubulin. Okay, so the proteins that are forming the microtubules, they are called tubulin, okay? And so microtubules, if you are asking one function, you can say that for formation of what? For formation of spindle. So the microtubules, they form the spindle fibers, but they will be organized by the what? By the centrioles. Okay, so formation of spindle fibers, fibers during cell division. Like I can say cell division. Okay, but specifically it's the nuclear division. Okay, Let me be very specific. Nuclear division. Okay. And then they also form the word, the formation of the word, formation of the flagella and cilia. Okay. Okay. Now, if you take the cilia, let me draw something like this. You see, if you take something like paramecium structure is something like this. Okay, and then so like say something like that. Okay, and then we have this nice hair-like structures. Okay, okay, here like that. It's okay. So hair-like structures. We also have cilia. So this is cilia all around the structure of paramecium. Okay. We also have cilia in our trachea, the windpipe, that trap the fine particles of the. Okay. All right. Good. All right. So there are cilia all over. Okay, great. So this cilia structure here is coming from the word, the cilia and the flagellum. Okay, which are these are structure. You know, remember stem cell has a flagella. Okay, so that flagella will be coming from the word, will be coming from the microtubule. So the cilia and flagella, they use the microtubule to form them. Okay, great. Then let's talk briefly about, should I talk about actin filament? That's okay. Well, I'll just talk about one, just one of them, just one. Okay. So the actin filament, you can see, okay, so when it comes to the actin filament, okay, one thing about it is that, sorry, let me talk about the intermediate, okay? So the intermediate is, from the name, you can see that the structure is in between the microtubule and then the word, and the microfilament, okay? Okay, but this one is quite, we have a lot of them in our body. In fact, there are a lot of them. Do you remember that I made mention of the lasmin? Do you remember lasmin? Lamin, not lasmin, lamin. Do you remember I made mention of lamin? Do you remember? Do you remember where I mentioned it? Do you remember? If you go back to do you remember where I mentioned it? Please, do you remember where I mentioned it? Do you remember? Okay. Do you remember where I mentioned it? Or you forgotten? Okay. You see, I told you that when I was talking about a nuclear pore, I told you there is this special protein structural protein within they help to control the nuclear pore. In fact, so in the nucleus, there is this protein we call the lamin. Okay, and if you remember, I made mention that this lamin, there is a mutation. I even mentioned that mutation. Okay, mutation in the word, in the lamin will lead to a disease we call progeria. Please do you remember? And please do you remember? And then I remember I brought you this picture. Let me take the progeria. Okay, like this. So these are the kits that I'm bringing the picture of those kids that tend to age quicker. Okay, so something like this. This one. Okay, so the kids that tend to age quicker. This disease is called progeria. This one. Okay, they tend to age quicker, and it's because of this. So this particular one like this, lamin. The lamin is a type of intermediate filament. Okay, all right, great. There are a lot of them, but this is basic cytology, so I wouldn't bother your head so much about other ones. Yeah, there's desmonin and there's vimentin, plenty of them. Okay, all right, good. So then we have what? Maybe the last one here. So let me pin this one here. The last one is the actin filaments. Okay, so the last one, the actin filaments. Actin filament is particularly important. Okay, these are small, so they are the smallest smallest. Okay. Okay, great. And this actin filament, remember, they help to even stabilize what we are calling as ectoplasm. You see, if you take something like amoeba and co, so they have this actin filament that tends to help to stabilize the ectoplasm. Okay, but the one you see, this actin, I don't know whether you've heard of it before. This actin, remember, in the muscles, okay, during muscle contraction, there are two fibers in the muscle. Okay, we have actin filament and what? And myosin. I don't know whether you still remember it. Old school muscle, so from the muscle. Okay, the SSD muscle. So if you take the muscle structure, like the ultra structure of the muscle, we have this structure, we call it what? We have something, okay, do you know what? Let me bring a picture so that you see rather than imagining it. Okay, let me see, sarcomere. Okay, so sarcomere. Okay, so let me bring it so that you see the structure I'm referring to. Okay, okay, so good. Okay, let me take this one here. Okay, 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 good. So let me copy this one here. So let me bring it. Okay, good. Now, so in the muzzle, okay, so in the muzzle, you see the structure. In the muzzle, you see the structure. This structure is called, that's the basic unit, the unit structure of the muscles, the sarcomere. Okay, so this sarcomere, you can see that we have the big structure here, we call myosin. And then you see we have this small structure, we call it what? We call it actin. Okay, so there's a thin filament. So this is the actin filament, this small one. Okay, so during contraction, what do? They slice over each other. Okay, so they slice over each other to bring about contraction. Okay, all right, so they slice over what? Over each other to bring about the contraction. So you can see that, so in the muscle, so this is the sarcomere is the basic unit of the muscle. Okay, all right, this is the sarcomere. That's the basic unit of the muscle. So with this sarcomere, okay, with this sarcomere, all right, the sarcomere like that, okay. So with this sarcomere, we have the actin filament here, and then the myosin. The myosin is the big one here. Okay, so this actin filament and the myosin, they slice over each other, and once they slice over each other to bring about what? The contraction. All right, good. Okay, so this one aid in what? So in the muscles, they aid in contraction of the muscles. Okay, and then another thing about another interesting thing about this one is the fact that they are also during the cell division. So when you see, when we talk about cell division, there are two things that I talked about. So you see cell division, when you talk of cell division, we have what? So when it comes to cell division, okay, we have two things. We have the nuclear division, we have the nuclear division, and what? And cytoplasm division. Cytoplasm division. Okay, division. And cytoplasm division is called what? It's called cytokinesis. Okay. Cytokinesis. All right, that's the cytoplasm division. Now, in animals, when animal cells are divided, okay, what will happen is this. Let me draw something here. What will happen is this. During the animal cell division, this is the cell. This is the cell that is going to divide. Okay. What will happen is that there will be accumulation here. Okay, accumulation on this side here. All right, of the actin and myosin, like that. Okay, and then this, they will go and pinch together like that. So there will be something we call ring of what? Ring of actin filament will form at the, let's say the nucleus has finished dividing like that. So one nucleus there, one nucleus here. Then, okay, what will happen? This actin filament here, the actin filament here like this, will do what? So sorry, this is the my nucleus. Let's say the nucleus has finished dividing, the nucleus has finished dividing. So there will be a ring of what? Actin filament, okay, at the equator of the cell, and then they will pinch inward. Okay, so this ring of actin filament, they form something we call cleavage furrow. Okay, they form, so what is here? This one, so the ring of what? Ring of, okay, of what? Of actin filament, okay, okay, form something you call the what? The cleavage furrow. Okay, all right, good. So you know what? So this one is specifically for animal cell. So here, this actin filament, okay, this actin filament here, all right, they can also, they were so in the way in cell division, okay, or let me make it in the word cytokinesis of what specific word of animal cells. The actin, this actin filament or the intermediate filament form what? Form, okay, so filament form a ring of what? A ring called what? Called cleavage furrow that divides the cell into two. All right, so here you see, so this cleavage furrow will pinch inward and eventually divide the cell into two. Okay, so this cleavage furrow will pinch inward and eventually divide the cell into two. All right, great. So the other functions, but these are two main ones that you need. Okay, yeah, great. All right. So we have the one. So we have the actin filament or the microfilament. They are what? They aid in they work in conjunction. So work. Let me say, actin filaments work in conjunction. Okay, in conjunction with the word, with the myosin, okay, to bring about contraction. Okay, great. All right, and then we talked about the cytokinesis in animal cells. Okay, they can form ring of what? So they form a ring called what? Cleavage furrow that divides the cell into two. All right, so this is what you can see for the cytoskeleton. Okay, please, any person so far? Any person so far? Any person so far? Any person so far? Before we move on to the two important organelles, okay, great. All right. Good. Now, once there's no question, let me move on to the mitochondria. Okay. All right. Okay, great. So all these things, you have them already, so you can easily refer to them. So I'm going to talk about mitochondria. So as you are, let me just go down. Let me go down and then talk about it here. Okay, mitochondria. Okay, great. Okay, mitochondria. We've mentioned mitochondrion. That is a singular. Okay, so that's a mitochondrion. Okay, great. That's a singular. Okay, good. So later, well, I think I will try and make a sketch here. All right, so that when I make the sketch, you also make the sketch as well. Okay. Oh, maybe I should just bring picture. Okay. Right. Okay, so now let me bring pictures so that we don't waste so much time. But in exam, I can ask you to draw. Okay, I can ask you to draw mitochondria and label the parts. Yes. Do you know what? Let me draw. Okay, I'll bring a picture, but I'll still draw it. So while I'm drawing, you also draw. Okay. All right. So that you learn how to draw. Okay, good. I am still old school when it comes to science. Okay, so this one here. Okay, all right. So let me put this one down here. But do you know what? I will still make a sketch of it. Okay, I'll still make a sketch. I know why I'm doing it. Okay, good. So you see, let's take it this way. So we have this mitochondria. So let me draw. I'm going to pick my marker here. So I'm drawing my mitochondria here. So you see, this is my mitochondria. Okay, let's say if I draw the mitochondria this way. Okay, like this, like that. Okay, okay, great. So this mitochondria, so it's double membrane bound. Okay, so double membrane bound, meaning that so oh, my mitochondria is not nice. Let me draw it. Let me draw it like this. Okay, then draw another line here. All right, good. Okay, good. All right, something like that. And then, so the inner part of this membrane, okay, so the inner part here, okay, you see, please, as I draw, please draw as well, okay? That's why, even though the diagram is good, but deliberately I'm drawing, so I expect you to draw as well. You should know how to draw it. It's very, very important to me. Okay, good. All right, maybe something like that. Let's try. Okay, good. So this is my mitochondria that I've drawn. And okay, so with this mitochondria, let's see. So we have the what? We have the inner membrane. So this part here is the inner membrane. Okay, this one is the inner membrane. Is that not it? So we have the inner membrane. Okay, so this is the inner membrane. This, so that you can see that the inner membrane is folded. Okay, so this is my inner membrane. Okay, membrane here. And the one here is what? The one here is an outer membrane. But do you know something? Let me draw another line here. Okay, so that you get a rough idea. I'm drawing another line deliberately. Okay, so let's see. Look at me. Okay, so I'm deliberately drawing another line here so that I'll be able to illustrate what I wanna talk about. Okay, okay. Okay. Okay, good. So in between the inner membrane and this, so this is the inner membrane. Let me put I here. Okay, let me use different color. Okay, so let's say I here, that's inner membrane. And then the outer one, let me put outer membrane here. So it has double membrane bound. Okay, good. So double membrane bound. And so in between the inner membrane, so here, this space here, there's a space. Okay, so this is the inner membrane, outer membrane, and then there's a space between them. So that space is called a what? So this space here, let me call this space here, the space here, okay? So let me put different color. Okay, let me bring different color here. So this space here, okay, the space between the inner membrane and the outer membrane, all right, this space here, okay, will be called what? Membrane what? Intermembrane space. intermembrane space. So that is here. So that's a so inner membrane, outer membrane. So the inner membrane is highly folded, okay, like that, and then the outer membrane is here. So in between the two, in between the inner membrane and outer membrane, we call it what? Intermembrane space. Okay. Oftentimes when we ask people to draw, people think that the beautiful nature of no, we are marking concepts. So you should be able to, I should be able to see those various concepts that I want there. Okay. So the inner membrane and then the outer membrane. Okay, great. And you can see there are pores in the outer membrane because certain proteins will move from the cytoplasm into the mitochondria, and then some will also leave the mitochondria. Okay, good. Then still we have the word. So inside here, inside this more or less the cytoplasm of the mitochondria, so we can get our DNA. Now the DNA here is circular, okay? So we have a circular DNA, okay? All right, circular DNA, okay? So we have the DNA here being circular. So we have so this is the circular, okay? So it is what? So it has a circular DNA, okay? Circular DNA, okay? All right, DNA. All right, okay. This DNA thing, let me underline and remind you, we'll visit it. There's something I need to talk about this DNA. All right, and then so the inner folding. Now these inner foldings are called what? They are called so singular is called crisp. Okay, crisp. That's for one. But when I point two of them, okay, if I point two of this folding, that's Christy, and Christy, you just add the word E to it. Okay, when you add E to it, the A is silent. So you have the Christy. Okay, so this inner membrane, they are folded to provide what? So so let me start typing a little bit. Okay, so all right, then let's see the ribosomes. Okay, so we have ribosomes in there. Okay, so we have the ribosomes. Okay, ribosomes. Okay, so we have let me point here. So we have the ribosome here, ribosome. Okay, all right, good. Good. So now, yes, please.
With the intermembrane space,
Mm-hmm.
is it not in between the matrix?
The matrix is here. Here is the matrix. What is here is the matrix. So is the matrix. No, forget about this drawing. They use one line to indicate the inner membrane.
Oh, okay. Okay.
They use one line to indicate the inner membrane. But I use two lines. It's a structure. So structure, we don't use a line. We use the two lines to indicate it. Okay. All right. We don't, like technically in science, we don't use one line to indicate a structure. We use two. Okay, so that is it. So here, so inside here, the liquid component here, which I call more like the cytoplasm of the mitochondria. Okay, this liquid component here is the matrix. So that's the liquid component, that's the matrix. Okay, so we have our circular DNA in there, we have our ribosome in there, all right, and a lot of enzymes are there. Okay, so let me type a little bit now. So, okay, this one, what happened? Okay, so let me bring this here. Okay, let me bring this one somewhere here. All right, okay, so that I can use this. Okay. Okay, why is it not coming? Let's see. Then let me try it. I can copy this. Copy, bring it here. Okay, good. So yeah, so something like this. Now, so let me type here. So now we have the word. So so key message here. We know the mitochondria. Okay, so they arise according they arise. So mitochondria, they arise, okay, as a result of what? As a result of endosymbiosis. They arise as a result of what? Endosymbiosis. Okay, as a result of endosymbiosis. Okay, all right. So I think we've talked a lot about this one already. And then so they have what? They have a double membrane bound, double membrane bound. Okay, or they are okay. So okay, let me make it. You know, I'm making it singular. So it is a double membrane bound, okay. It's double membrane bound organelle. All right. And then what's also with this double membrane bound, which means that we have inner membrane and what? And outer membrane. Okay. All right. And you know that our inner membrane is what? Is folded. Okay. To form the what? To form the cristae. Okay, the cristae. Okay, and what is the cristae doing? Okay, that provides a word. That provide cristae is plural. Okay, if I add it to is plural, that provide large surface area for enzymatic activities. Okay, so there is enzymatic activities. So that's what the cristae is doing, providing a large surface area for enzymatic activities. Okay, good. That's the work of the cristae. All right, and then so we have the outer membrane. So the outer membrane serves as a barrier. So the outer membrane with channels. Okay, with channels. Okay, all right. Serves as a barrier between the what? Between the mitochondria and the what? And the cytoplasm. So that actually regulates movements of substances there. All right. And then so in between them, so we have the inner, and then we have the what? We have the inner, and so we have the inner, and then we have the what? The outer. Okay. So inner here, outer there. All right. Then in between them is the what? Is the intermembrane space. So in between them. So in between the inner and outer, okay, is the what? Is the intermembrane space. Is the what? Is the intermembrane space. All right. Is the intermembrane space. So in between there is the intermembrane space. And this intermembrane space is very, very important. Okay. That is where hydrogen ions, in fact, that is where hydrogen ions or protons, hydrogen ions accumulate. Okay, that is where hydrogen ions accumulate, okay, during a process we call, during the word electron transport word chain, okay. So in the electron transport chain, we have the hydrogen ions, okay, hydrogen H+ ions were accumulating, accumulating, right, during the word electron transport chain. All right, good. And then we have the what? So we have the liquid component here. Okay, the liquid component, which is the matrix. So the matrix is more or less, I'll call that one the more or less the cytoplasm of the mitochondrion. And this matrix, that is where the enzyme, a lot of the enzymatic activity, that's where they occur. So that's a more or less the liquid portion. Okay, portion. Okay, containing enzymes and okay, containing. Okay, good. Containing enzymes and enzymes and stuff like that. Okay, so here two main processes tend to occur here. I think I said this some of them the last time. So the two main processes that tend to occur here, we're talking about the word the Krebs cycle, okay, inside the matrix, and then the word. So the Krebs cycle okay inside the matrix, and then within the word the inner membranes and the membrane chain, we can talk about the word the electron transport chain, okay, which we shall talk about it maybe later, slightly a little bit detailed. Okay, all right, good. So we have the Krebs cycle here. So the intermembrane, like those intermembranes, the inner membranes. Okay, the inner membranes will have the word. Do you know what? Let me bring you a picture that will make it easy. So I wouldn't explain so much here like that. So let me show you a picture of it, of what I'm talking about. Okay, good. So here, if you take this one, okay, let's say, let me duplicate this one here. If I duplicate this one, okay, now let me bring this one, okay, let me duplicate, okay, great. All right, let's see something here. So here, okay, so you can, all right, so you see, this is what is here. So this is the intermembrane chain that I was talking about. So this is the mitochondrial structure. So you can see this is the cytoplasm here. This is the intermembrane space. So you can see that the intermembrane space is, as I told you, it's accumulation of hydrogen ions here. Okay, plenty hydrogen ions are here. And this is the inner membrane. So the inner membranes are the inner membrane here is where the electron transport chain are actually located. Okay, so those proteins forming the electron transport chain. Okay, so the electron transport chain here. Okay, all right. So during the process, so let me this chain. Let me put it here. Okay, in a folder. Yeah, correct. Okay, so within the inner membrane, so within the membrane itself. Okay, so let me add that one too. So within the inner membrane are the what? Are the electron carriers. Okay, so we have other protein, we call them protein complexes and mobile electron arrays. Okay. All right. So here we have, let me minimize this one for small. Okay, great. All right, good. So we have on this one, so here you see these ones say 1, 2, those big structures here. Okay, these are protein carriers. So you see in the inner membrane, we have these are we call them protein complexes. So these are the 1, 2, 3, 4. Okay, 1, 2 here, 3, 4. Okay, these are protein complexes. And these ones here, this one we call this one ubiquinone, and this we call it cytochrome. Okay, we have this one we call it cytochrome C. Okay, and we call this one ubiquinone. So these one you don't need these intricate details now. Okay, So we have these ones here. So these are mobile, these two here, they are mobile electron carriers. And in the inner membrane here, we have the one, we have the protein complexes. So if I'm being a little bit specific, I can say that there are four protein complexes and 2 mobile electron carriers. Okay, all right, good. So these are in the inner membrane. So these ones, they are the one that will carry out the electron transport process. So here, so this is the, so you see the electron transport. The electron will pass on from here to there. This, what we see here is what we call the electron transport process. And in the course of that, they use the energy to pump hydrogen ions here. So this is the intermembrane space. So this intermembrane space here, that is where the hydrogen ions will be pumped. Okay, so they pump the hydrogen, so they accumulate here. You can see that there are 4 plus hydrogen, 2 plus hydrogen. So these are, and then what will happen is that after they pump this hydrogen, so let's take it that the energy in the electrons will be used to pump hydrogen here. Okay, already there are more hydrogens here, but they will use it to pump hydrogen ions against the concentration gradient here. And so when they do that, when they pump the hydrogen ion against the concentration gradient, then at the end of the day, this hydrogen ion will pass through this ATP synthase. ATP synthase is like a turbine. So this ATP synthase, okay, this ATP synthase, the hydrogen ion will flow through the ATP synthase, and during that process, they will convert ADP to ATP. And remember, ATP is the energy for the cell. Are you with me? Let me take it one more time. So what will happen is this. So here, you see in the inner membrane, okay, in the inner membrane, we have this these 4 protein complexes and 2 mobile electron carriers. And these ones, what will they do? So they carry out something we call the electron transport chain. So electron transport chain is basically this electron released here will be passed on from what? From this complex to the next complex, to that, like that. So from complex 1 all the way to complex 4. And that passing of the energy of the electron is what you call electron transport chain. And as they do that, the energy released by the electron is used to pump hydrogen into this space here. Okay. And so at the end, after the whole process, remember I told you the last time that this particular process, before even the Krebs cycle will start, okay, there should be oxygen there. So you see at the end of the electron transport chain, somebody should be there to capture the electron. So who will be there to capture the electron? It is the oxygen. So oxygen will be there to capture the electron and turn. So during that, and remember when electron is added to a molecule, it is called what? Reduction. So that electron will be used to what? Reduce oxygen into what? Into water. Okay, so they will add hydrogen into it, and then water will be formed. So that is what happens. So there should be, during the electron transport chain, there should be oxygen there, okay, to capture the electron. If this oxygen is not here, the process wouldn't, okay. Do you know why? Because this electron here is a highly reactive electron. The moment it leaves the cell, it's a radical, it's a free radical. It can go and destroy our DNA, it can destroy phospholipid, it can destroy proteins. So somebody should be there to capture it. So during the electron transport chain, okay, this is where, okay, the electron. So this one here, this one, the NADH is electron carrier. So when this NADH, they will form this NADH during the Krebs cycle. So during the Krebs cycle, NADH and FADH2 will be formed during the Krebs cycle. So when they form it during the Krebs cycle, all right, when they form it during the Krebs cycle, then they will come and release the electron, okay, to the electron transport chain. And the electron transport chain within the inner membrane, there are 4 protein protein complexes. That's this 1, 2, 3, 4. And then 2 mobile carrier. This is one, ubiquinone and cytochrome C. Okay, here. And so they pass on the electron. So the fourth one, after the fourth one, then the electron will be captured by the what will be used to reduce oxygen into water molecule. All right. Then after that process, the energy as the electron is being transferred. Okay. All right. So maybe I need to write too. First, I don't know whether you can put all this stuff in your head. Let me duplicate the slides here. Okay. All right. Let me add some text here. Okay. So we say that so in the matrix, okay, Krebs cycle. So in the matrix, what will occur? Krebs cycle will occur. Krebs cycle will occur. All right. So in this Krebs cycle, when this Krebs cycle occurs, okay, so the Krebs cycle will occur. And this Krebs cycle will produce what? NADH. All right, and what? And FADH2. All right, and these are electron carriers. Okay, so they carry, these are electron carriers. Okay, so they carry electron carriers. Okay, all right. So when they get to the world, when they get to there, so they will carry this high energy electron.
Doc.
Yes, please.
Doc, please. What are the actual names of the electron carriers? Like the N for what? The NADH, N for A for. What's the actual name?
That's the hydrogen nicotinamide adenosine dinucleotide phosphate. Dinucleotide.
The N is
nicotinamide.
nitrogen, right?
This is hydrogen. So here we mentioned hydrogen first. So hydrogen nicotinamide adenosine dinucleotide. You don't need to worry your head about these names.
But why are they written like that?
You asked for it. Get it.
My head is spinning.
That's why I saved you.
Circular motion.
You know something? One good thing about science is that some of these acronyms, so like you ask people exam question and then they just need to write ATP and they are fine. Some they want to show that they know. Then they go and write adenosine this thing,
Triphosphate.
Yes, adenosine triphosphate, and then they will make the spelling, and then we'll mark them wrong. Meanwhile, if they wrote ATP, you're fine. Yeah. So please don't let your head spin. God is with you. So what will happen is that during the Krebs cycle, okay, the Krebs cycle occurs in the matrix, all right, and in the Krebs cycle they will produce what? They will form this molecule, and this molecule they are carrying high energy electrons. So you can see from my diagram here. You see, look at that. You see that when it gets there, this NADH will release its electron, all right, and when it releases its electron, they will use it to pump the hydrogen up. Then when it gets to this one, the FADH2 will also release its high energy electron, and then they will pass it on, okay, to the complex 3. So complex 2, it will release it to complex 2. Complex 2 will pass the electron and to the cytochrome, okay, to the mobile electron carrier. This is the ubiquinone, and then we'll pass it to the 3. Okay, so they release their electron, the electrons that they've captured, the energy in the form of electron. They go, and when they release it, they use that energy from it to pump hydrogen into the what, intermembrane space. So after this pumping of hydrogen in the intermembrane space, so here, all right, so other word, so in the inner membrane, they are what? There are 4 protein complex, okay, complexes, and 2 mobile electron carriers, okay, which are this small c and that, okay, and they perform the what? The 2 of them, they perform what? Perform the electron transport chain. Okay, so when they perform the electron transport chain, remember the energy there, the energy there is used to what? It's used to pump H plus what? Ions into the what? Into the intermembrane space. All right, so they pump it here. Good. After they use it to pump this, so at the end of the distance, at the end of the process, remember, electron will where? The electron will be captured. Okay, good. The electron will be what? The electrons will be what? Will be captured by the what? By what? By oxygen to form water. So you know where the water is coming from, right? You know where the water is coming from after, you know where the water molecule is coming from after what? Respiration. Okay, good. It is coming after the what? After the electron transport chain, the high energy electron will be captured by the electron, and in presence of hydrogen ion will be reduced to form what? Water. Okay, all right, great. Now, have you seen this hydrogen ion that is pumped here? Now, this hydrogen ion will now move freely. Okay, so the hydrogen ions up here, the hydrogen class ions in the what? In the intermembrane space, all right, or in the intermembrane space will flow, okay, through the what? Through ATP synthase. Okay, this ATP synthase is acting like a turbine. Okay, turbine, remember turbine, when the turbine is spinning, then you are generating energy, right? Okay, we form the ATP synthase. So as the hydrogen is flowing through this one, then it is turning the turbine, converting ADP into what? Into ATP. So they will add, okay, during that process, they will add what? One phosphate, we call it inorganic phosphate. They will add 1 inorganic phosphate to the word. So this is adenosine, okay, dinucleotide phosphate. All right, and then hello.
Yeah, please. I would like to ask a question.
Please go ahead.
When the hydrogen ions or the electrons are transferred, added to the oxygen to form water. Is it that all the hydrogens are not transferred to form water?
No, not all of them.
Oh, okay.
Yeah,
so,
you can
see, the remaining ones are sent to.
Yes, so there's some. You see, the thing that a lot of this process, a lot of them are okay. You know, look at plenty of them are on this cell. So definitely look at that. So this one is breaking, giving two hydrogen. So definitely there are some hydrogens here, but just that they keep pumping them. And you see, I didn't say channel, I said pump. Why? Because they are using energy. And where is the energy coming from? The energy is the energy in the electron. So the energy in the electron is what is being used to pump the what? The hydrogen ions into the intermembrane space. So still there are some hydrogens here. Still there.
Okay.
Okay, good.
Okay.
Okay, so what is the name of this side? What's the name of this side? This side is what? This side of the this side is what? Where I'm pointing of the mitochondria, this side will be what?
Inner membrane.
The inner membrane is this. This is the inner membrane. But what is this side?
That would be the matrix. The matrix.
Good. Well done. Good. So that would be the matrix. Well done. So here, there's the matrix here. So this after the electron transport chain, then this high energy electron needs to be captured. Okay, so it will be captured in presence of hydrogen, and then it will be converted. It will reduce oxygen into water. Now this high energy that has been pumped here, okay, that this energy, this hydrogen that they pumped here, okay, what will happen is that then it will flow through this ATP synthase, which I refer to as like the turbine of the cell. As they flow through the ATP synthase, then they convert the word ADP into ATP. Okay, so that's the adenosine dinucleotide phosphate into adenosine trinucleotide phosphate. And so this is how energy is made in the cell. So the electrons, so this flow of what? The flow of the hydrogen ions through the synthase, it is called what? It is called chemiosmosis. Chemiosmosis. Okay. So the flow, this flow of what? Okay, the flow of the hydrogen ions through the ATP synthase, okay, through that turbine, okay, to form the what? To generate the ATP from ADP and the P, okay, that process is called what? It's called chemiosmosis. So together, so inside here, we call it what? We call it the whole of this process. In fact, the electron transport chain here, okay, which occur in the inner membrane, all right, and then the chemiosmosis, okay, which involve the flow of the what? The flow of the hydrogen through the ATP synthase to form the ATP. These two, so collectively, okay, electron transport chain plus the what? Plus the chemiosmosis, you call that one, okay? Okay. You know what I'm talking about. I don't want to waste time. Chemiosmosis, we call that one what? Oxidative phosphorylation. Okay, good. You see, there is phosphorylation. Phosphorylation is the addition of the word of the phosphate to the ADP, okay, to form the word, to form the ATP. That process is called phosphorylation. But in this particular case, you can see that it occurred as a result of what? It occurred as a result of oxidation. Okay, it occurred as, so we call this one oxidative phosphorylation. Okay, so it occurred as a result of what? As a result of losing of what? You know, moving electron from, okay, losing electron. That's the oxidative stuff. And at the end of the day, the energy was pumped, you know, they used the energy to pump the hydrogen ion, and then hydrogen ion flow through turbine, okay, generating ATP. All right, beautiful. So now I've given you overview of what happens in the mitochondria. Okay, so let's see the general function of mitochondria. Okay, so general function of the mitochondria. But before that, remember, yes, please.
Is it necessary for the water to be formed or released before the excess hydrogen forms the ATP?
Please come again.
I wanted to find out if it is necessary for water to be released or formed before ATP is synthesized.
It doesn't matter. This process we are going, they occur almost synchronously. This is just one of it that we've described, but it doesn't stop. It is still going on. So it doesn't matter. Do you understand that? It's not stopping. The thing is that we've just shown just one, but it is not stopping. This process is still ongoing. It's not stopping. So do you understand that? It is continuous process. Because if it stops now, all of us will be cackers. So it doesn't stop. It's still ongoing. So when the water is forming or this, it doesn't really matter. Because the process is not, it's a continuous process. It's not stopping. So water could be here. It doesn't matter. It doesn't change anything. Do you understand that? Whereby maybe to work and to stop for this one to start. No, it's not happening like that. It's a continuous process. This one is going, this one is going because the cells are just doing it to release energy quickly. If not, we will all be cackers. And you see, if you look at what I gave you earlier on, you remember that when I talked about a cellular respiration, okay, you see, remember that when I talked about when I made mention of cellular respiration, cellular respiration, okay, respiration, okay, cellular respiration, okay. Remember I mentioned these 3 steps. Remember that cellular respiration, I said it involves what? Glycolysis, followed by what? Krebs cycle, okay, Krebs cycle, followed by the electron, okay, the electron transport chain. This is what we talked about. Okay, great. And you see all these two, this one doesn't require oxygen. No oxygen required. No oxygen. Okay. But these two, oxygen must be present before it will start because there should be oxygen to catch the electrons. So right after the Krebs cycle, and the Krebs cycle, here, even Krebs cycle, ATP is produced. Okay, so in Krebs cycle like this, we produce what? We produce ATP, okay, but the major product that will be produced is NADH and what? FADH2, as well as what? ATP. But ATP the ATP are not much. All right, this is what is produced here. Then this high energy molecule containing high energy electron will now be sent to the way. Will be sent to the inner membrane where it will carry out the what? The electron transport chain. Okay, that is the oxidative phosphorylation. Okay, all right, okay. So here is where the electron transport chain will occur because they are what? They are protein complexes. Okay, protein complexes. Okay, so we carried out by the protein complexes and then the mobile electron carriers. Okay, like that. So they will do the electron transport chain, mobile electron carriers. Okay, they will do the electron transport chain. Then the energy, the energy during the electron transport chain is used to pump hydrogen into the intermembrane space. Then this hydrogen will fall through the what? Through the ATP synthase, converting it into ATP. So here, large amounts, in fact, large amounts of what? ATP are produced during the electron transport chain. Okay, I mean at SS level you tell the electron transport chain, but at the university level you know that there are 2 processes that tend to occur there. You have the electron transport chain plus the what? Plus the chemiosmosis, and that collectively give us the what? Give us the oxidative phosphorylation. Okay, phosphorylation. That's it. Okay, good. All right. So here, now that we know this overview of that, okay, what is the function of mitochondria? What is the function? What's the primary function of mitochondria? First. Please, what's the primary function of the mitochondria?
Produces energy for the cell.
Produces energy.
So you know that, okay, so here it is the word. So it's a site of energy production. So it produces what? Produces energy, okay? Energy produces and stores, okay, and stores energy in the form of what? In the form
ATP.
in the form of what? ATP. All right, good, excellent. And you know how the processes are involved. Okay, beautiful. All right, but some of the tricky questions. Let me ask you this question. Let me ask you this. If I say that all ATPs are produced in the mitochondria, true or false? All ATPs are produced. It's false. ATP can be produced during glycolysis. So here, the last one I gave so here, during the glycolysis, you spend 2 ATP and produce 4. So you have net ATP. So you have 2 net ATP here. And this ATP is produced. Hello?
So only Krebs cycle occurs in the mitochondria.
Both Krebs cycle and electron transport.
Okay.
These two. Okay. So
but
the Krebs cycle,
glycolysis does not occur in the mitochondria, right?
but I didn't mention mitochondria. You didn't hear the question that I said. I said that all ATPs are produced by the mitochondria. True or false? That's the question I asked. And not all ATPs are producing. Glycolysis will produce ATP. It is producing ATP outside the mitochondria. So bacteria doesn't have ATP. It doesn't have a mitochondria, but it can generate energy through glycolysis in the cytoplasm. Okay, so that makes that statement false. Okay, good. But the vast majority, okay, in the process, the vast majority of ATP, okay, are produced from the what? From the aerobic respiration. Okay, that will involve. So the aerobic respiration will involve the Krebs cycle and the electron transport chain that tends to generate large amounts of ATP through something we call oxidative phosphorylation. Okay, the ATP production here we call it substrate level phosphorylation. Substrate level phosphorylation. Okay, phosphorylation is basically addition of phosphate. Okay, all right, great. This one is phosphate level, substrate level phosphorylation, and then the one in the electron transport chain is what? It's oxidative phosphorylation. Okay, all right, good. So yes, please.
Please, so in a semester exams, can we be asked to describe the Krebs cycle?
No, I wouldn't ask you here.
I
won't ask you here. You need to know about it, but I won't ask you here. There will be a time I'll ask, but not this one. Not here. The thing is that I will, the next blog, I will teach you some aspects. Okay, I will teach you some aspects, and then we'll go into detail of Krebs cycle. But sometimes when I'm going into that detail, people don't get it. People miss it. And the reason they miss it is that they didn't get the foundation here right. That is why I've mentioned it here. So that it will be in your head that after glycolysis, Krebs cycle, electron transport chain. Okay. But in that time, I'll go into intricate detail. But because I know I will be teaching you that one, that is why I want you to put this one. You need to get the basis here right. Okay. Once the bases are there, like you know where, after glycolysis, you know that the Krebs cycle will occur in the matrix. Okay. And then generate what? Generate this. okay, generate the main products are these two, and then ATP will be produced as well. Okay, so once the foundation is there, it is easier to build upon. That's why I'm going to this level. Okay, all right, good. But I won't bother you about it now.
Okay.
Doc, Doc,
Yes,
Yes.
please.
Doc,
Yes,
I
please.
wanted to find out
please,
if,
please, I
Mhm.
beg you. So far as you've mentioned that we shall do it once again in the next block. In a simple term, Krebs cycle is what? Is about what? In a simple
It's a
term, so that
cycle. It's a cycle that generates this. It's a cycle, like the way we have it's a reaction cycle. Okay, it's a reaction cycle. Okay, let me just show you quickly. Like this. So let's say, let me. Okay, good. Okay, like that. Okay, so have you seen it here? So here, this is the Krebs cycle. Okay, so these are the various reactions like this. All right, and then the two, you can see that if you look on that, you can see NADH is produced here, NADH is produced here, and FADH2 is produced here. Okay, so NADH, 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 and then FADH2. So these are the main things. In ATP we produce as well, but it's just one. Just one cycle. In fact, there are two Krebs cycles. Okay, that is generated from one glucose, but okay, it produces just one ATP per cycle. So the two will produce two. Okay, good. So here, so but the main products are the NADH. So this is the cycle. Okay, this is the cycle. But at this time now, it's basic, so I won't worry your head. But I'll worry your head sometime next block whereby but you need to get the basis. You need to know the basis here where it is occurring. It's occurring in the mitochondria matrix. You should know what the mitochondria matrix is. Okay, and then the main products are there. It's okay, great, so that you can easily build upon. All right, good. Yes, please.
Yes, sir, please. So in the next block, would we be talking about uric cycle so that I put aside my question?
No, please.
Oh, okay. Then I wouldn't ask at all because I wanted to ask whether under the uric cycle there is electron transfer as well.
Then you're asking now.
No, in fact, uric cycle, I don't want to divert our class a little bit because that's quite intricate detail. Okay, I can go there as well, but that's a little bit complex to divert our class a little bit. Okay, I think after class, I wouldn't mind to explain to you. Okay, those are basic stuff. Okay, good, yeah. So one is what? So reproduction and storage of what? So it produces and stores energy in the form of ATP. That's the principal thing that it does. Okay. Then what else again? Another thing that mitochondria is also involved, which we hardly talk about, is the way. You see, have you seen the programmed cell death? The programmed cell death. What's the term for programmed cell death? Yes. You remember the programmed cell death? Well, there is a term for it. Okay.
Apoptosis.
Oh, good. Well done. Okay, well done. Proud of you. So yes, so the programmed cell death, which we call what? Apoptosis. Okay, good. So so they also, so then once you people know it, then let me go. Okay, apoptosis. Okay, good. So they also involve in what? They also involve in, they regulate what? Cell death. Okay, programmed cell death. Okay, so they also regulate, they also involve, in fact, they are much more involved in programmed cell death than even the lysosomes. Okay, so when we die, that is what happens. In fact, when cells are going to die, that's basically the ribosomes, some proteins, enzymes inside the ribosome will be released, and then we call something caspase. Okay, the caspase will be released into the cytoplasm, and then it will trigger some processes. Okay, death. Okay, so a whole lot of complex processes, and eventually our cell will die. Okay, so that is it. So they are actively they regulate the word. They also involve in the word regulation of a word, regulation of Program cell death. Well done. That's good. Of apoptosis. Well done. Well done. Proud of you. Okay, number 3. Okay, so regulation of let me, in case those who makes that class, that's the program cell death. Okay, that is the apoptosis. Well done. Okay, good. And then do you know what now, please, anytime you put your hand in your armpit, what do you feel there? Please. Or when you put your hand in your armpit, what do you feel?
Heat.
Under your armpit, what do you feel?
Heat and heat.
Heat, right? Okay,
Yeah.
good. Heat and what? Hey, what do you want
Heat
to
and
add?
scent.
Heat and okay. Yeah, the men. Yeah, it's true. That one is important. Okay. Yeah, that one. True men. You have to have strong one. Okay, that's a sign of a true man. Good. Well done. Good. Okay. Now, so where is that heat coming from?
I think it is coming from through metabolic activities.
Okay, great. Now, so that heat is generated. You see, we have this heat generation. So we have this sugar. Then we have this fat. Let me call it fat. Okay, so we have this brown fat. Okay, and this brown fat, some of them are so under our armpits. A lot of them is accumulated there. Okay, especially babies. Okay, babies on their neck region. And you see, good. Now let me come here. Great. So there is a special channel. Okay, I won't go into much detail of it now. Okay, great. There is this special channel. Now, during when the temperature is high, okay, when the temperature is high, you know, let me show you a picture of it. Okay, let me show you a picture of it. So, okay, once I've talked about it, let me show you a picture of it. Mitochondria. Yeah. Let me show you a picture. Let me see. Can I see a picture? These are very complex ones. Okay. All right. No, I'm not getting a picture. I'm not really getting a picture. Okay, good. I've gotten something here like that. Okay, good. Okay, I've gotten a simple picture here. It's loading. Let me show you it. It's a very nice one. Okay, good. So here, there is this one here. Let me put it on this side here. Oh, good. I've just covered everything. Okay, good. So let me shift this one slightly to the side. So on this mitochondria here, okay, on the mitochondria, you can see that we have ATP synthase, right? That when the hydrogen ion is going down, what would they do? They convert the ADP into ATP. Now, during thermogenesis, that's heat generation, what happens is that there is this special structure here. There's this special channel, okay, on the inner membrane, okay, and this special channel, where am I writing my functions? Where am I writing them? Okay, good. There is this special structure here. We call it what? We call it UCP. Okay, 1. Okay, good. So there is this channel, we call it UCP1 here. Okay, UCP1 here. And this UCP1 channel, what it does is that instead of this hydrogen ion to go through the ATP synthase to generate ATP, if I want to generate heat, the hydrogen ions will go through the UCP. And as they go through the UCP, okay, then heat energy is generated. Okay, all right, good. So that's what happens. So here, so if so in babies like this, okay, and under our armpits, okay, you see that heat energy is there. Okay, you see anytime you touch your armpit, you have heat. And then the baby, so the babies like that, the babies tend to have a lot of what? They have a lot of brown fat on their body. And so this brown fat tends to generate ATP. That is why people are very big. They tend to generate a lot of heat on their body. Okay, it's those the Obushi people. Okay, Obolo. All right, the Obolo people tend to generate a lot of, because the fats will have a lot of brown fat. And so this brown fat, what happens is that they have this. Okay, so this is the mitochondria will have UCP channel on them here. okay, on the mitochondria, on the inner member of the mitochondria. So what happens is that this hydrogen ion sometimes can pass through the UCP, generating heat, but it's not in every cell. Okay, so for babies like that, they have a lot of the brown fat, okay, and then that actually generates what heat for the body. So here, mitochondria can also be involved in what? In heat generation, okay, which is termed as a word, chemogenesis. Chemogenesis. All right, okay, good. Okay, great. All right, so I will just give this one as the main functions of the mitochondria. Okay, good. Then now you see, when you come to this mitochondria, there's another very important thing we need to talk about here. Okay, so let me duplicate this slide. Okay, all right. Then let me clean this one here. Now, which cells? Okay, which cells? Okay, so my question is, which cells? Let me put it here. Which cells will have a lot of mitochondria? A lot of mitochondria. Okay, which cells? All right.
Muscle cell.
Yeah, muscle cells. Good. Okay, but there's a champion. I want you to mention the champion first before we talk about muscle cells. You are right, muscle cells, that's correct. But there is a champion. Okay, you have to mention the boss first. Which one? Okay, sperm, almighty sperm cell. Okay, so sperm cell, in fact, it is the mammalian word, mammalian cell with a mammalian cell with the highest number of word, with the highest number of mitochondria. Okay, so remember when you can, okay, we shall come to sperm very soon. Okay, so in fact the sperm cell is the mammalian cell with the highest number of mitochondria. Good. Now we can talk of the muscle cell. Okay, muscle cells, okay, because they need energy for contraction. Okay, all right. Okay, and then what else again? You know, those cells that are involved in what? Involved in active transport. Okay. So cells that are moving molecules by active transport mechanism. And then anytime we talk about active transport, that's moving molecules against the word, the concentration gradient. Okay, moving molecules against the concentration gradient. Okay, so cells that are actively involved in what? That are involved in active transport. Okay, that is you also have what? They have numerous mitochondria. So good. What else again? So muscles. Okay, so muscles, both the skeletal muscles and then the heart muscles, will all have a lot of what? Mitochondria. Okay. Then another very important point that I want you to know here. Okay, you see that. Let me bring this one here. Okay, let me take this one. Okay, another very important point that I want you to know here. Okay, so let me bring this one. Okay, so let's see. Okay, let me take this one. We are done with this. Okay, so let me bring this one here. Now, you realize that mitochondria has what? Oh no, look at me. Sorry. I've put it in the wrong place. Okay, so let me bring it here. Okay, let me bring it here. All right, great. So if you have something like this mitochondria, you can see the mitochondria contains what? DNA, is that not it? Okay. Now, if you take the sperm cell, this is very important point we need to make here. Okay, so let me come in. Even though we'll come to sperm cell very soon, but let me draw this one quickly. If this is the sperm cell, if we take the sperm cell like this, okay, all the mitochondria, so this is the head of the sperm cell like that, and then the TPA, okay, that's the acrosome. Now all the all the mitochondria are here. So this side is where the mitochondria, all the mitochondria are all hiding here. Okay, we have this ring of mitochondria here. Now, during fertilization process, this mitochondria here will not enter the tail. Now, during the fertilization process, this is the egg. Okay, the sperm will come here. The sperm will they will dissolve. The acrosome will dissolve. When it dissolves, then it's only the head, only the nucleus here will enter the egg like this and go and fuse to form the white, to form the zygote like that. So this tail region will not enter. All right. So what does that mean? What it means here is that all the mitochondria we have in our body, okay, the mitochondria we have in our body is coming from who? Our mother or our father? Yes.
It's coming from the mother.
It's coming
from the mother.
Good, excellent. So all the mitochondria we have.
And so there's a question to that.
Okay, let me land, and then you ask the question. So all the mitochondria we have in our body, okay, because in the fertilization process, the tail breaks off. This tail will break off like this. So there's no mitochondria from our dad. So the mitochondria in the sperm, all that it does is to facilitate the movement of the sperm to the egg. That's all that is, to provide energy for the sperm to get to the egg. That is all. All right. The moment it gets to the egg, it is done. It is done with its work. So during the fertilization process, this tail breaks off, and it breaks off with all the mitochondria. So because of that, remember this mitochondria contains DNA. Okay. So let's take it this way, that I am at the hospital. Some people have mixed up their babies. So right now we've established the fact that all the mitochondria we have in this cell here, all the mitochondria we have in our cells are coming from our mother. Okay, our mom. So you and everybody, you have the same mitochondrial DNA like your mom. So when you take them, this DNA in the mom, because the DNA from the mitochondria in the father will not enter the egg during fertilization process. All the mitochondria we have is coming from our mother. So our mitochondrial DNA, we have the same mitochondrial DNA as our mom. So for instance, for Kwame here, so Arabe's mitochondrial DNA is the same mitochondrial DNA I have in me. So which means that my aunties and my uncles from my mother's side, so I'll have the same. So let's take it this way. I have one uncle called Kwesi. So I have the same mitochondrial DNA with Uncle Kwesi. Why? Because Uncle Kwesi is a sister. Uncle Kwesi's sister is Araba. Okay, and they all come from the same mother. So my grandmom passed on their mitochondrial DNA to my mom. Let's say I have another auntie here, Auntie Equia here. So Auntie Equia here, like this. Also, I have the same mitochondrial DNA, because the same one that my grandmom passed on to Araba is the same one they passed on to my uncle, the same one they passed on to my auntie. So my auntie, my maternal aunties, those my cousins here. Okay, let's see, that my cousins here. Okay, Naomi, all these people here, I have the same mitochondrial DNA. So if I'm the one here, I have the same mitochondrial DNA. Why? Because my grandmom passed this mitochondrial DNA to their mom and passed it to my mom. The same mitochondrial DNA, okay, the same mitochondrial DNA here will go into what? Antiquia, and Antiquia will pass it to my cousins. Okay, but you see, my cousins, my uncle's children, I will not have the same mitochondrial DNA with them, because they will pick their mitochondrial DNA from my uncle's wife. Okay, like that. So I won't have with my uncle, but I will have the same mitochondrial DNA with my maternal cousins. Okay, my maternal cousins. Okay, my maternal cousins, those like coming from my aunties, not from my uncles. Okay, all right, good. So this is so when babies mess up at the hospital, how would you identify them? Let's say the babies, they mess up at the hospital. Let's say people have given birth, they mess up their babies. So if they call geneticists like me and I can, what I would do is that I will just check the mitochondrial DNA in the child and check the mitochondrial DNA in the mother. Okay, all right. So here, this is a very important note. Okay, please. Sorry, please. I'm in a Zoom meeting. I'll call you back. Okay, please. I'm in a Zoom meeting. I'll call you back, please. All right. So so with this one, okay, what is going to happen is this. So we have this mitochondrial DNA. Okay, so all that when the people mix up their babies at the hospital, okay, what will happen? You check the mitochondrial DNA to identify them. So the point I want to make here is that mitochondria DNA is inherited, okay, maternally. All right, okay, all right. In sexually, okay, reproducing organisms. So when people mix up their babies at the hospital, the best thing to check is to check the what? You check the mitochondrial DNA, okay, because okay, the mitochondrial DNA will be the same as is the same as. Okay, so please you can go ahead and ask your question, please. Please ask your question.
Sir, thank you very much. Now we have it now in our mind that the mitochondria generate energy in the form of ATP. Okay, sir, please, so this kind of energy that we are talking about, is it the energy that energy is the ability to do work? So the energy as in in carrying out all day-to-day life activities, that is one. That is one of the questions that I want to ask. And also, if now we've learned that the mitochondria that we have is coming from our mother, does it mean that if someone is weak or the energy level of someone is low, then the mother is responsible for that?
You can't say mother. Okay, well, let me ask. So yeah, so the energy we are talking about is coming from our mitochondria. This is so we have
don't bring anybody.
We don't have.
Don't bring anybody.
So we don't have this thing. You have various types of energy. So you have chemical energy. So this ATP we are talking about is a form of chemical energy. So when you break ATP down, it releases energy. So our muscle contraction. So you see energy. So you see we take in food. That's the glucose. The glucose. So it is the glucose that will undergo the glycolysis first. Okay. So the glucose. Okay. So here, this cellular respiration here. Where is it? So the glycolysis here. So the glucose here during the cellular respiration, we have the glycolysis. So the glycolysis here, okay, was split. So glycolysis is the splitting of glucose into what? Into pyruvate, two pyruvate molecules. Okay, and this pyruvate will then be converted into acetyl-CoA. Okay, so this pyruvate, okay, if there is oxygen there, it will be converted into acetyl-CoA. And to this acetyl-CoA, that will enter into this Krebs cycle. If you look at the Krebs cycle, you can see that you say glucose here, later. Have you seen that this glucose is becoming acetyl-CoA? Okay, so it is this acetyl-CoA that will enter the Krebs cycle. All right, good. And so when it enters the Krebs cycle, the energy will be donated in the form of what? The electron energy will be carried by this one. And then when it gets into the electron transport chain, it will be released and then become as hydrogen ions. Then when it descends, when it flows through during the chemosynthesis, then it will use it to generate ATP. So after the generation of ATP, for instance, if the muscles want to contract, the contraction of the muscle, it is just basically ATP breaking down. When it breaks down, the ATP breaks down. Okay, it's generate the ADP and P. And during that process, high amounts of energy is produced within the cell. So that's a chemical energy. So, but the glucose is too big. So the glucose needs to be converted into a currency, energy currency, that is manageable by the cell, and that energy currency is the ATP. Okay, so they convert, so the glycolysis, the Krebs cycle, and electron transport chain, it's all go through so that they generate the energy in the glucose in the form of, it will be generated into the ATP, which is the manageable energy currency of the cell. Okay, so that energy currency is a chemical energy. And then the second question is a very good question. The second question is that, so you asked a question that if you are having an issue with your mitochondria, doesn't it come from the mother? That's true. There are a lot of genetic disorders, especially heart issues, okay? There are a lot of genetic disorders, okay? Genetic disorders that we experience, okay? All right? And then they are coming from our mother, a lot of them. And because we inherited from our mom, okay, most of the times, any disease, so that is why when you are there, I always tell people that, you see, mothers are very, very important. Okay, I mean, I'm not saying fathers are not important. In terms of health-wise, health determination, whatever that happens to your mom, will invariably, in terms of the those things like mitochondria, like for instance, mitochondrial diseases, and there are a lot of them. There are a lot of mitochondrial diseases. Okay, we have the MELAS, we call something mitochondria and cephalomyopathy. Okay, it's actually, this one's like that. It has something to do with those heart and our muscles and stuff like that. Okay, all right. And all these activities, okay, all these diseases, all right, they are actually affiliated to mitochondria. So any problem, any genetic disorder in relation to mitochondria is coming from your mom, because you wouldn't pick mitochondria from your dad. So sometimes if your mother is complaining of heart issues, okay, or certain issues pertaining to the heart, pertaining to the muscles, all right, you should also know that if you are not careful, one day you will suffer from it. Okay, because it's what?
So
come again.
relation to the heart, as in blood pressure, BP.
Not necessarily blood pressure. I mean, we have a lot of heart disorders. I'm talking in relation to mitochondria. Okay, for blood pressure, it's not only for mom alone. If your dad also has it, we can. I mean, those are okay. We can inherit it in both ways. Okay, but what I'm saying here is that not BP in particular. Though we have some sort of genetic disorders that are in relation to mitochondria DNA, all right, or they are in relation to just mitochondria. And because all the mitochondria we have in our body is coming from our mother, okay, the mitochondria you have in our body, they are coming from our mom, then, which means that whatever sort of problem your mom is having, if it is relation to mitochondria, you should know, so that you know how you go by your day-to-day life. Because there are a lot of mitochondria disorders. And so, not necessarily, so sometimes, some of them are in relation to the heart. Okay, there are a lot of them. And so, when you have problem with the mitochondria, so you have to know what killed your grandmom. Okay, if you have idea of it, they should tell you. If your mom complains about some issues, you should know. Because there are some of the, like mitochondria alone, and there are so many disorders that can be inherited from the mitochondria. Okay, all right, please. Please, I'm in a meeting, so I'll call you back, please. Okay. I'm in a meeting, I'll call you back, please. All right. So there are so many. This thing. There are so many disorders that are associated with mitochondria, and so once you know what is happening to your mom and stuff like that, it helps you, okay, so that certain things you need to avoid. Okay, that's what I'm trying to say. Okay, good. So there are a lot of mitochondrial disorders, okay, that some of them are associated with the heart, some of them are associated with the muscles, okay, because remember, mitochondria is going to generate energy, okay, for all these structures. So if there is any genetic disorder in relation to them, okay, even the brain, even in our brain, you know, the brain cells, okay, they need a lot of energy, okay, so our brain cells and stuff like that. So if there is there is some mitochondrial disorder, it even gives some sort of mental health issue, Okay, there's some mitochondrial disorder that they can result in what mental health issue. There's some mitochondrial disorder that can be like stroke. Okay, all right. So some can even affect eyes, your vision. All right. So there are a lot of them. Okay, there are some mitochondrial disorder that can act like epilepsy. Okay, so we have quite a lot of them. So that is why you need to know what if your mom is complaining of something. Okay, in my case, Araba complains of hypertension. So and then Kofi also complains of hypertension. So which means that genetically, I'm genetically predisposed to be hypertensive. But do you know what? I will not suffer from hypertension. Do you know why? Because the things that will fall hypertension, okay, I don't keep it. When I got to know of they being genetically, which I'm genetically predisposed, no, I don't watch horror movies. Any scary thing, I don't watch it. If somebody does something that annoys me, I blast the person. We end it there. So I don't have to keep any grudge anywhere. So I'll be thinking that then my heart is pumping. No. We blast you. We get it. Life goes on. Okay, so that I don't have to ruminate about anything. There's something that's bothering me. No. I have to be free, man. Okay. So these are some of the things. Once you have the knowledge of something, it helps you to avoid certain things. Okay. All right. Good. So yeah, so from this one here, we should know of, so this one, we know that mitochondrial DNA is inherited maternally in sexually reproducing organisms. Okay, that's very, very important. And then my last question for you is that—
Hello, sir.
Yes, please.
I just want to ask this simple question. Based on this slide that is on, it says that the sperm cell is the highest number of
contains the highest number of mitochondria.
Okay, and I just want to ask that we have a heart muscle cells, don't we have that?
Yes, sperm has more.
So since the heart works non-stop, can't that one be the highest one instead of the sperm cell?
No, it's not. It doesn't. Things don't work like that. You can see the sperm cell. Look at where the sperm cell will be deposited. Okay, yeah, the heart muscle has mitochondria, a lot of mitochondria, all right, but the sperm cell has the highest.
Oh, okay.
Okay, looking at where the sperm will be deposited, the sperm will be deposited in the cervix. Okay, let's say in the vagina, let's say somewhere in the vagina region. In the vagina region, when they're deposited in the vagina region, they have to go over the cervix all the way, climb the uterus, move all the way to the fallopian tube. Now remember, this is just a cell. So the cell climbing that hill is like you climbing Mount Everest.
Okay.
Have you seen it? So
hello?
It's hello.
Yeah, Doug.
Yes, please.
Yeah. Since you made mention that we have a lot of mitochondria in the brain cell. And people, even though I've not read much about it, but they've been saying that the children, our intelligence is gotten from our mothers. So is it related to this mitochondrial thing?
For intelligence, you get from both. It's not necessarily from our mothers. It's good.
Thank you for that. Thank you for that.
Okay, you get from both. It's not necessarily from our mothers. Okay, it's genetic. When you come to genetics, I think I will explain that one. Okay, we get from both. It's not necessarily our mothers. Okay.
Thank you for being both.
If you are smart, I mean, there's high possibility of giving birth to smart kids. Okay, it's not only to the mother. If the mother is smart, it helps. Okay, and stuff like that. Okay, like for instance, you can see that teachers naturally, teachers' kids naturally tend to do well. Okay, all right, and you see, it's not necessarily because the teachers are smart, but it's also due to the fact that teachers are more inclined to have more books at home, okay, to help kids to study. So naturally, okay, research has shown that teachers, like kids whose mothers are teachers, are most likely to be doing very well academically. Okay, but you know that the intelligence is genetic, and you inherit it from both parents. Okay, all right, good. Okay, so my question goes, my last part of the question, do we have some cells that do not contain mitochondria at all? Okay, so cells that do not, that don't contain what? Mitochondria. Can you give me an example of those cells as well?
So can it be red blood cell?
Yes, excellent. Okay, so red blood cell like that, you see in the red blood cell doesn't contain mitochondria. Okay, so it's good that way, so that it doesn't consume the oxygen it is transporting. Okay, so the classic example, EG is the word EG is the word red blood cells. Okay, so red blood cell is transporting the oxygen. So if we're transporting the oxygen, we don't expect you to consume it. So red blood cell like that, it transports what? Oxygen. So it doesn't carry mitochondria because it doesn't use oxygen. Okay, so it's respired by what? By glycolysis. Red blood cell will work. So red blood cells they respire by what? By glycolysis. So they rely on what? They respire by what? Glycolysis. Okay, so red blood cell, the red blood cells, they don't what? They do not contain mitochondria. Okay, all right, good. Any headache so far? Any headache so far? Okay, any headache so far? Okay, please let me speed up a little bit for the next 5 minutes and talk about chloroplast quickly. Okay, chloroplast. All right, quickly so that then I'll end with it. Okay, great. So let me go here and then okay, great. Chloroplast. Okay, so chloroplast. All right, so when you come to the chloroplast, okay, the chloroplast is remember chloroplast also they arise as a result of what? Arise. Okay, arise as a result of what? As a result of endosymbiosis. Okay, endosymbiosis. Yeah, as a result of endosymbiosis. Okay, great. And then I don't think I'd like to draw chloroplast here. Okay, great. But let me bring some sort of image. Okay, let me bring a simpler image of it. Simpler. So chloroplast. Chloroplast. Okay, let me bring some simple diagram of chloroplast. So that's okay. All right. So let me take something simple like this. Okay. Let me take something simple like this with nice diagram. Okay. So let's say my chloroplast is here. Let me bring this one here like that. Okay. Great. Okay. So the chloroplast. Okay. Remember the chloroplast is they all come as a result of endosymbiosis here. In this case, like that, the liquid component. So it's also, it has what? It has double membrane bound. Okay. It is what? It is double membrane bound. It is double membrane bound organelle. Okay. Double membrane bound organelle. Okay. And then the liquid parts of this one, as the liquid parts of the mitochondria is called matrix, the liquid parts here is called stroma. Okay, it's called stroma. Okay, so the stroma is the what? The liquid. Okay, the liquid part is the liquid part of the what? Of the chloroplast, okay, is the liquid components of the chloroplast. Now, with this liquid components of the chloroplast, okay, good. Now, when it comes to the chloroplast, okay, so they also have a circular DNA, just like mitochondria, circular DNA, okay, like mitochondria, and they also have the what? They have the ribosome as well, ribosome, okay, ribosome, just like mitochondria, okay, good. Circular DNA. And then ribosome, just like mitochondria. All right, good. It also has a outer membrane and inner membrane. Okay, that's good. Now, you see, so inside the mitochondria, we have this structure. Okay, these rod-like structures. These rod-like structures are called what? They are called as a thylakoid. Thylakoid. Okay, they are called thylakoid. So this one. So this rod-like green structure, they are called thylakoid. And this thylakoid, that is where the photosynthetic pigment, that's where the chlorophyll, that's where the chlorophyll okay, is located. Okay, that's where the chlorophyll is located. Okay, so inside the thylakoid, okay, that's where the chlorophyll is located. Or in some, if you want to be a little bit specific, you can say that they are the photosystems. Okay, the photosystems. Okay, systems are located. So they capture the sunlight, okay, for the photosynthesis to trigger off the photosynthesis. Okay, and then, okay, so let me say chlorophyll, maybe photosystems. Let me put it that way. Okay, yes. Okay, good. Allocated. So these ones, they capture the sunlight. Okay. Now, when you come to photosynthesis, okay, when you come to photosynthesis, we have two parts of photosynthesis. We have light phase of photosynthesis, and then we have what? The dark phase of photosynthesis. The dark phase of photosynthesis is called Calvin cycle. Okay. Now, this light phase of photosynthesis occurs in the thylakoid, where the photosystems are located. So the light phase, where this is where the light, it will capture. Okay, that's light dependent phase. Okay, light dependent phase. Okay, light dependent phase. Okay, and then the dark phase is simply light independent. Whether light is there or light is not there, it will still occur. All right, light independent phase. And this one, so here, the light phase, okay, is light independent, and it occurs where? It occurs in the thylakoid. Okay, the thylakoid. Okay, and then the dark phase, okay, which is called Calvin cycle or light independent, okay, all right, phase. This one occurs in the what? In the stroma. In the stroma, the liquid part here, this liquid part, okay, the liquid component here is the stroma. So that is where the dark phase of photosynthesis, which we call the Calvin cycle, okay, in the what? In the stroma. All right. Okay, good. So so we know that cells, plant cell, one of the unique structure of the plant cell is the possession of the what? Is the possession of the chloroplast for what? For photosynthesis. Okay, so plant cell has the what? Has the chloroplast as organelle. So here it is found in what? So this organelle is found in the what? In the plant, okay, plant cells, okay, plant cells, okay, plant cells, and then some green algae, okay, so green algae, okay, some algae, almost all the algae they have it, okay, so the algae, okay, so the algae, okay, so the alga cells also have the word, the chloroplast, so that they are able to carry out photosynthesis. Okay, now in plant cells, some of the cells have a lot of them. Okay, so if you take the plant cell like this, okay, if you take the plant cell like this, there's one type of cell we call it the palisade. The palisade cell, okay, palisade cell. Okay, let me show you a picture of the palisade cell quickly. Let me say, let me go to the inner structure of leaves. Okay, so structure of what? Of the leaf. Okay, structure of the leaf. Okay, all right. Let me. So let me bring this one for you here. Okay. Now, if you take this one here, if you take the one, okay. Okay, good. Okay, so let's see something like this. Something like this here. Now you can see these cells here. These cells here, this region is called palisade mesophyll. The cells here are the palisade cells. So these palisade cells, you can see all these dots this around are all where? Okay, they are all chloroplast. So the palisade cell contains what? Large number of what? Number of chloroplast chloroplast. Okay, contains large number of chloroplasts. Okay, and then so look at here. So the policy says a classic example of plant cells with large number of chloroplasts. Okay, because it's going to absorb sunlight for photosynthesis. Okay, great. But we can also get cells plant cells that do not work. For instance, if you take something like some plant cells don't have chloroplasts. For instance, the cells here, the cells of the epidermis. Look at this cells here. These cells of the epidermis here. Okay, they don't have the chloroplast. So except gut cell has chloroplast, but the other cells of the word. So cells of the word of the epidermis do not don't have chloroplast. All right, the cells of epidermis, they don't have chloroplast. Look at that. So on top of this leaf here and the one here, except gut cells. Okay, except gut cells, but if not, all the cells of the epidermis they don't. So likewise, so cells of epidermis, they don't have the cells of epidermis don't have chloroplast. And then root hair cell also doesn't have root hair cell it is in the soil. It's not going to absorb sunlight for photosynthesis. So also doesn't have chloroplasts. Okay, good. So on this note, we still need one more meeting. Honestly, for us to finish the pace that I'm going, which is good for your learning sake. So because of the slow pace that I'm going, we are still behind. Okay. So today is what? Today is, what class do you have again today? The chemistry people have another class, right?
Yes, sir.
Yes, please.
What time is your class? It's 11:30, right?
Yeah, 11 to 4.
11:30 to what?
4 o'clock.
To 2 o'clock.
Yes.
4 o'clock. Okay.
We have
4 o'clock. 4 o'clock.
4 o'clock. Is it possible? Can we meet? Yeah. Can we meet again today in the evening? Okay. In fact, I need to go somewhere, but I will cancel it. Okay. So that you see, we are moving in a very nice pace. So you guys are going to teach our children, and so I have to make sure you master everything very well. So that when you go, you can teach them the way I'm teaching you. Okay, that you can go to class without book and you kill everything. Then I'll say, yes, that's my people. You know, like Angela will kill the whole class. You know, kill the quality and people. Yes. Mm-hmm.
Some centers don't go into details as we are doing.
Don't worry about it. I'm the course coordinator. Don't worry about that. Okay.
Doc.
Yes, please.
Hey, Doc.
Yes, please.
Monday, we don't have any class. Biology people. Can we put it on Monday?
But I heard some chemistry people are part of your class. So.
Okay, yeah, we are here.
Okay, Monday. Are you guys not going to school Monday?
We can do it for today. We can do it 4 o'clock.
Yeah.
Okay, but 4 o'clock, the chemistry people will be too tired. The chemistry
okay, so we can go to 5.
Okay. So Monday what time? In the morning.
Monday in the morning.
Early in the morning.
Yes.
Okay, so please. Monday. Okay, so let us agree. I can run with you and we'll finish, but that is not what I want because I know you are going
we
to
don't want that.
What the pace we are going is excellent. Okay, this pace we are going is good. Okay, so that you master everything. Right now, go and take any question and you will kill it. Trust me, you will kill it because we are just touching almost every part and stuff like that, which is very, very good. Okay, so what's happening is that Monday. Okay, so let's decide it quickly before our chemistry people run away. All right, so Monday. Okay, good. 7:30. Monday. Okay, no, Monday because it is new week. Okay, people are a little bit tired. Okay, and stuff like that. So let's make it 8 o'clock.
In the morning.
Okay. Yes, 8 o'clock in the morning. Okay, 8 o'clock.
Okay.
8 o'clock in the morning. Monday I'll be very, very busy, but I'll try and then I'll come because Monday our first year students are coming on campus. So
Okay.
yes, so I'll be slightly busy. So we have to do it. In fact, if you're able to do 2 hours, I'll be glad. Okay, so Monday, yes, please.
What about the next day?
That we will do it. So
Okay.
that's why I'm fixing this one so that we'll be able to. So Monday, God willing. So Monday, 8. Okay, so Monday, 8 o'clock. 8 o'clock. In fact, realistically, so Monday, 8 to 11. That's more realistic. Okay, I will try 8 to 11. And then the Monday, what we are going to do is this: we will do the specialized cells. Okay, and then after the specialized cells, so please in your free time try and read something about it. Specialized cells, that is so you go for sperm, nerve cell, sperm and nerve for animals, nerve cells, okay, and then maybe try and then add adipose cells, okay, and then read something about chief cells, chief cell, and then parietal cell. Okay. So here, but the main one you need to focus is this one, sperm and nerve. This is the one you need to focus on, sperm and nerve. But I will ask you questions on this one too.
So we don't have the price for this one. Oh.
Don't worry, I'll send this one to you. I'll send it. I wanted to add true or false questions, okay, but I couldn't do it. That is why I've not sent it. But I will send it this one today. All right, good. And then after this one, so this is what we'll do on Monday, God willing. And then we'll add this one to it. We'll add the transport in cells. Okay, in cells. All right, great. Okay, so this is what we'll be doing. So Monday, 8 to 7 a.m. All right, good, sharp. All right, yeah, we will be on fire. Okay, so we are going to finish these 2, and once we are done with these 2,
But this is 8 to 7.
Yeah, 8 to 11. Sorry, 8 to 11. So we'll try and finish specialized cell transport mechanisms in cells, and then we'll add maybe cell division, cell cycle. Okay, so this is our target. Okay, if we're able to finish it, we'll start something on molecular. Okay, we'll start something on molecular.
But sir, please, so
DNA.
sir, please, so I learned after today we are left with one online lecture. Then we'll go for practicals, and then I think quiz and exams begin.
For biology, you won't have practical for biology. We need to meet 4 times. We need 4 meetings. We've met how many times? Is that the 3rd one?
This is the 3rd one.
Okay, good. Don't worry. We'll fix it. I'll finish it. I'll make sure we'll finish it. We can use Monday. I will squeeze Mondays, okay? And then I'll finish it. Don't worry.
Thank you.
Hey, Monday. Hey, Monday is what? This coming Monday, I have a lecture in the morning. Can
we do it in the evening?
No, let's do it this evening. Please, let's do it this evening. So let's take it this way that my chemistry people will finish their class. My chemistry people will finish their class on 4:30, right?
So, sir, can we fix it at 6 o'clock to 9 o'clock?
6 o'clock to 9. 6 o'clock to 9. 6 to 9.
Yeah.
Okay, 6 to 9.
6 to 9.
Okay, don't worry.
Yeah, I think that one will be louder.
Please. Okay,
so that
then
we
what
can
about
finish
our
the
people are going to do the cooking? Oh, please, so forgive me, please. Okay.
We will manage.
So, sir, they should try.
Oh,
Right
we
after
manage.
here, right after here,
People
they should.
should help. We should try. Help us.
Oh, that's good. That's good. They will
So
help.
let's
We'll help
help
you.
us and finish fast.
We'll help. We'll help. Yes, we'll help. Okay. So we'll try and fix it at 6 to 9 p.m.
9. Yes,
Then
sir.
we'll try and cover this. Okay, and possibly some aspects of this one. All right, and then we'll have one more meeting. Okay, so our practical time for biology, we'll use it for the genetics. That one, I think I didn't send so much of genetic slides to your this, so I will try and combine the whole class for the genetics one. Okay. That one,
Okay, but so if we're able to finish today,
yes,
the specialized cells, then maybe today's week
yes,
we'll start with the what do you call it,
genetics.
the genetics, and
Okay,
then we'll continue using the practical
good.
session to
Good.
finish the
Excellent.
genetics.
Excellent. 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 Okay, we can do that. So we'll make sure we'll move with the speed of light and nail these ones. Then, okay. But the genetics is there. You
Sure,
need
sir.
more. The genetics is there because that is where things we will say a whole lot of naked things. You know what I'm saying? Okay, naked. You know what I'm saying? Yeah, man. Yeah, man. You know the spermatogenesis and eugenesis. You know what I'm saying? My ladies, are you there? You need that class badly. Okay, good. So the genetics, everything's going to be on point. Okay, you know, Kwame is geneticist. So even if non-genetic stuff I'm teaching in this video, you can imagine what I'll do with the genetics. Okay, good. So I wish you all the best. Have a very good morning, and then have fun, and then may God richly bless you. Okay? And I fish too.
Sure,
All right.
sir.
Okay. I pray that
Happy
next year by
Sabbath,
this
sir.
time, some of you have started your masters, and then wrapping up the masters and running for the PhD as well. Okay. All right. God bless you. And have a lovely
Yes, sir.
Amen. Amen. Amen.
Amen. You'll be contacting
Thank you.
you for that.
Oh, sure. Why not? Okay. You know,
So we'll be calling you for guidelines on that too.
you know
We'll be
that
calling
you
you,
know that my ladies, I'm always there for you. Okay. The men, I think you guys on your own.
Yes, please.
Okay.
You are.
They spread, they
Men,
spread.
Papa, men, they talk of Papa.
The men you're on your way. All right.
Man, do man.
Oh, wait till man do
Wait till man do man.
You see that? All right. So
Man don't suffer. Man don't
yeah.
suffer.
Oh, Charlie, how you gonna do? So please, this evening we are meeting. Please don't miss this class. I'm begging you, don't miss this class because you know I'm supposed to go somewhere in the evening. I'm canceling it so that we'll be able to have the class. Okay. All right, good. So please try and then come. So
Rest assured.
all right, thank you. Okay.
Hello,
So
Doc.
hello, please. Let me hear from you, please.
Please, would these meetings outside the scheduled time also be recorded to our YouTube?
Yes. Yes. So I will speak to Becky. I'll speak to Becky so that Becky will get it recorded for us. But please try and take notes. Try and take notes as I teach, so that okay, after class, okay, you have on stuff. Okay, please. Okay, try and take notes. Okay, thank you.
It was never. It's not easy. Gradually, we get there.